Hello, I want to welcome everyone to our Insulin 2021 event, the celebration of 100 years of insulin therapy. It has been 100 years since the discovery of insulin changed the world for those with diabetes, from a sentence of slow, painful death to the opportunity for a healthy life. The researchers and clinicians of the Endocrine Society have been at the forefront of finding better ways to care for patients with diabetes. We develop strong relationships with our patients and look forward to improving their quality of life. We are advocating to work to break down the barriers to diabetes care and for universal access for insulin therapy for all those who need this life-saving medication. Throughout 2021, we have been highlighting the insulin's past and advances in insulin replacement we use today. In our program today, we are not only going to celebrate the past, but also many of the advances that future might hold in diabetes medications, insulin delivery advances such as artificial pancreases, use of artificial intelligence, and other technologies. Our goal today is to prepare you for this changing environment so that you can educate and treat your patients, as well as educate those who follow in your footsteps. We hope you leave today's presentation inspired to make a difference in people's lives. Thank you for being part of this journey. I'll turn it over to my colleagues now to kick off the event and for the introductions.
Thank you so much, Dr. Weisham, for that great introduction. My name is Colleen Williams, and I'm the Manager of Public Relations at the Endocrine Society. We want to thank our sponsor, Lilly, for making this event possible. For today's event, some sessions are pre-recorded. However, the Q&A portion with our speakers is live. My colleague, Jenny Gingery, who's the Director of Communications and Media Relations at the Society, will be facilitating the Q&A portion today. You may type your questions into the box at the right-hand side of your screen at any point during the webcast. Please be sure to indicate who your question is for. These presentations will be available on the Endocrine Society's YouTube channel and website following the event today. For our first session today on cellular and biological advances in type 1 diabetes, we are pleased to have with us Dr. Alvin Powers of Vanderbilt University Medical Center and Dr. Douglas Melton of Harvard University. Dr. Powers, we invite you to please speak first.
Thank you, Colleen, and thanks to the Endocrine Society for inviting me to participate in this exciting session celebrating the 100 years of insulin treatment. It's a great thrill to do this. It's a great program that's been outlined today. Here are my disclosures, none that are relevant to today's discussion. Here's what I'd like to do today. I'd like to first review the current understanding of the genetics and the pathogenesis of type 1 diabetes. And in this, I'm going to highlight gaps in our knowledge and some of the increasingly recognized heterogeneity in type 1 diabetes. I then would like to provide an overview of type 1 therapies that are on the horizon. And in that, I'd like to provide a context for the other speakers and the topics that are going to follow me today. And then finally, I'd like to discuss some considerations for the emerging, evolving therapies for type 1 diabetes that will be relevant to the discussions that follow today. So this slide shows the often used model of how type 1 diabetes develops. So on this axis, we have the amount of functional beta cell mass and time on this axis here. And the model goes that there is some genetic predisposition to type 1 diabetes, and that at that time, the beta cell mass is normal. Some unknown triggering event, thought to be some environmental agent, initiates an autoimmune process, and that this autoimmune process, shown by this bar here, gradually destroys the beta cells of the pancreatic islet. Now, what's been recognized over the past 5 years that type 1 diabetes has been reclassified into having 3 stages: type 1 stage, type 1 diabetes stage 1, type 1 diabetes stage 2, and type 1 diabetes stage 3. And in this phase of stage 1, there are 2 autoantibodies reflecting the autoimmune process, but the glucose tolerance is normal. In stage 2, these antibodies are still present, but there is dysglycemia and mild glucose intolerance. And as a rule, people are asymptomatic during this phase. And then the third phase is symptomatic type 1 diabetes. So one of the changes about how we approach type 1 diabetes, so we used to think the diagnosis was here, but now the American Diabetes Association, the Endocrine Society, the ASD now think that type 1 diabetes begins in this phase. So stage 1 and stage 2 are part of the disease. Shown at the upper part of this is a schematic of the pancreatic islet, with the beta cells in green, the alpha cells in blue, and the delta cells in purple. And so what's thought is that we go from a normal islet like this to there's a loss of beta cells, and then eventually all the beta cells, or essentially all the beta cells, are lost, and we're looking at an islet that has both alpha cells and delta cells, but very few beta cells. The vascularization and the innervation of the islet shown in this by these structures here appears to be present in all stages of diabetes. Now, these are drawn as very sequential serial processes, but it's likely that they are not so straightforward. And so while this is drawn as a smooth, never changing graph, it's likely that it may have stops and starts, but we really don't know what the longitudinal nature of this looks like. Now, I'm going to highlight 3 areas of this curve today. We're going to talk about this early phase and some of the genetics. We're going to talk about some of the autoimmunity. And finally, we're going to talk about therapies that might be used in stage 2 and stage 3 diabetes. So let's begin here. And what do we know about the genetic risk for that? Now, we all know, it's been known for long, that about 50% of the genetic risk is related to HLA genes. But what's becoming increasingly recognized from studies that were done initially in the UK Biobank, that many individuals, maybe up to 40% of individuals who develop type 1 diabetes, develop it over the age of 30 years. So this graph shows cases of type 1 diabetes based on age, and one can see that many of those cases are over the age of 30. So that raises a very important question. So one of the issues is emerging concepts that type 1 diabetes can appear at any age, but we don't know whether it's the same disease when it appears when somebody who's 32 as it appeared in somebody who was 5. And there is this growing sense that the autoimmunity is different in individuals who are young with onset of type 1 diabetes, say less than 10 years of age, compared to older individuals. And this really doesn't even address people who have type 1 diabetes that are in their 40s that initially appear like they have type 2 diabetes, so-called latent autoimmune diabetes of adults, where they have disease that is probably different, but we don't know that whether there are different pathogenic processes. We do know that the HLA risk factors are similar in all of those individuals. Another thing that I want to talk about in this before diabetes begins is the heterogeneity in beta cell mass. So this slide shows autopsy studies from the Mayo Clinic by Peter Butler and his colleagues looking at age. These are non-diabetic individuals and looking at their beta cell mass on the y-axis. And one can see that there's considerable heterogeneity in beta cell mass, and that some people may have 3 to 4 times the beta cell mass of other individuals, all of whom were normal and non-diabetic. So that raises a question: is how does that influence our risk of type 1 diabetes? So if you'll see this redrawing of the Eisenbarth model, where we have beta cell mass on this axis and time here. So if a person had, say, average beta cell mass here, their time to reaching diabetes here would be, but if they had, say, half of that beta cell mass, or they had twice that beta cell mass, their time at arriving at the clinical diagnosis of type 1 diabetes would be very different. So the baseline beta cell mass is likely very important in when and if someone develops type 1 diabetes. So how and when is beta cell mass determined? So I just want to show you just a couple of things that are work from colleagues at Vanderbilt University, Marcelo Brashova and Diane Saunders here, and a project that they're working on with the University of Florida, looking at the pancreas of individuals who are less than the age of 10. And they studied a large number of these, and they've looked at different features of the islets in there. And so, for example, here's the pancreas from a neonatal period. Here's one from infancy, and here's one in childhood. And one can see the islet stained with insulin glue in green and glucagon in red, that as the pancreas grows, these islet cells, which in this early period are scattered and are really not assembled into full islets, then become assembled into islets. And as the pancreas grows, they become spread out through the pancreas. Now, what's interesting about this in another levels, if one looks at the islets here in children at that age, their islets look very similar to adult human islets in that alpha cells shown in red and delta cells shown in blue and DAPI here are scattered throughout the islet. That's very different than what we see in young individuals here, where there's a central core of beta cells and the alpha and delta cells around the perimeter. These are much more reminiscent of mouse or rodent islets. So this tells you that there's considerable plasticity and cells are moving around and changing during this time period. So if one thinks about how this might influence our beta cell mass, here's a scheme showing beta cell mass here in age. So this is the individual who would have normal beta cell mass, and this is one who would have half beta cell mass, and this is someone who would have 2 times beta cell mass. But it appears that beta cell mass is heavily determined by what goes on in the first decade of life. So by the age of 10, our beta cell mass is determined. So this is an important determinant of whether or not someone develops type 1 diabetes and for type 2 diabetes. The other reason that this time period is very interesting are studies from Annette Ziegler and her colleagues in Germany, where they followed individuals from birth who were high risk genetic for type 1 diabetes. And what they did is they measured blood and collected a number of things every 3 months. And then they followed them to see whether or not they developed type 1 diabetes. And what they found, if you had no autoantibodies, that your risk of developing type 1 diabetes is very low. If you had 1 antibody, it was a little bit higher. And if you had 2 or 3 antibodies, your risk of developing type 1 diabetes in the next stage of life was quite high. But look when those islet cell antibodies appear. They appear in this very early part of life, which is the time that I've already shown you there's considerable plasticity in the human pancreas. So now let's talk a bit and talk about the autoimmune process when beta cell mass is thought to be declining. And what do we know about that? Well, we know that you can predict that. So this is work from TrialNet. Here's a map showing there, and there are centers in Australia as well. So whether or not you have 1, 2, 3, or 4 autoantibodies, your risk of type 1 diabetes can be predicted. So if you have 3 or 4 antibodies, autoantibodies, and you're a first-degree relative to someone who has type 1 diabetes, you have about an 11% risk per year in developing type 1 diabetes. So in these types of studies of first-degree relatives and other types of studies that are being done in the general population, one can predict the onset of type 1 diabetes fairly well in some, but not all individuals who ultimately develop type 1 diabetes. But one of the concepts that's really beginning to influence the way the field is thinking is this increasing recognition of heterogeneity in type 1 diabetes. In borrowing a phrase from the asthma literature, where there are different types of asthma, people have sometimes these are endotypes, where there are different forms of type 1 diabetes. And I want to just summarize on this slide some of the heterogeneity that's been recognized. First, there's immunologic heterogeneity, meaning that the autoantibody frequency, the profile, and the epitopes of those autoantibodies is variable from person to person. The response, type 1 interferon, innate immunity, and T cell signatures is different in certain forms of what looks phenotypically to be type 1 diabetes. There's also metabolic heterogeneity, so that the rate of decline in beta cell function and mass is variable in stage 2 and 3. Not everyone progresses to diabetes, and not everyone progresses to diabetes at the same rate. Another sign of heterogeneity. And this leads to a difference in the degree and duration of C-peptide production. We used to think that everyone was totally deficient in C-peptide and beta cells, but now it's become increasingly recognized that some individuals make C-peptide for a number of years at the diagnosis of type 1 diabetes, but it's variable. It's not true in everyone. And then finally, there's a response to the immunomodulatory therapy that's being tried in individuals with new onset type 1 diabetes. Another example of heterogeneity in type 1 diabetes refers to the insulitis, the inflammation that's in the islet. Both the degree and location of that differs between and within individuals. In a certain pancreatic section, you may see some islets that are totally spared and some islets that are completely devoid of beta cells. So what's responsible for that heterogeneity within an individual is not clear, but there's also difference between individual and individual. CD20B lymphocytes have been found by Sarah Richardson and Noel Morgan and their colleagues at Exeter to be much more common in individuals who are young when they develop type 1 diabetes. And then there's a difference in the degree of beta cell loss. And as we mentioned, how rapid that happens, there's heterogeneity. And then there are differences between insulin negative islets and islets within the beta cells. So this concept of heterogeneity is something that we should think about as we go through the day, because there may be some individuals where one therapy is preferred, another individual, because they have a different form of type 1 diabetes, another therapy might be desired. So I now like to talk a little bit about a brief overview of the type 1 diabetes therapies that are on the horizon to provide some context from the speakers who are going to follow later in this session. So we're going to talk both at individuals who are at stage 2 and individuals who are at stage 3. And when one thinks about the therapies that are here, they're really a competition of 3 forms of emerging and or evolving therapies for type 1 diabetes. And I'm going to show that. So here's this first form of exogenous insulin replacement. And we're going to talk a lot about that today, about how insulin modifications of the insulin molecule, how those are delivered, sensors, pumps, algorithms, AI algorithms, those sort of things. So that's one therapy. The next is cell-based insulin delivery. So the transplantation of islets or introducing cells, or stimulation of regeneration within the pancreas, or possibly transdifferentiation of host cells into insulin-producing cells. So let me just schematically show that on this slide, where these are beta cell replacement strategies here. And this is one where you might modulate the functional beta cell mass. If you remember, I said that some individuals have beta cells for an extended period of time after diabetes onset, and almost all individuals have considerable beta cell mass, albeit insufficient, when type 1 diabetes is diagnosed. So in terms of beta cell replacement therapies, we're going to hear from Dr. Melton after this about in vitro differentiation strategies to turn embryonic stem cells into insulin-producing cells that can then be transplanted. And of course, transplantation of human cadaver islets continues to be actively pursued and investigated. And it works quite well, but the issue is the limitation of the number of islets that are available. So those could be transplanted. And then on the other side of cell delivery would be, how do you modify the endogenous beta cell mass that's still remaining? Could you do things that might protect the beta cells from immune or metabolic stress and thus improve beta cell function? Could you stimulate proliferation? Could you stimulate, through neogenesis or transdifferentiation, more beta cells? Could you target things to the beta cell that might protect our immune or augment their function? And then the final would be protecting our immune modulatory therapies, where an islet shown here being attacked by either T cells, or by antibodies or T cells, that if you could use inventory modulatory therapy, you might be able to slow this process. And that's been shown by Kevin Harold and other colleagues. And this slide here shows the rates of developing type 1 diabetes. These are in individuals who are at stage 2, and they were given a tocilizumab versus placebo. And one can see that in this group, there was a prolonging, about 2-year prolonging of the onset of type 1 diabetes. So this is very exciting and suggests that individuals in stage 2, that the onset of diabetes could be prolonged. So how does this work? Can it be improved to completely stop diabetes? And what are the side effects of modulating the immune system with this one-time intervention? So where are we going from here? And I'd like to conclude by outlining some things that I suggest that we think about as we go through the day. And so for you baseball fans, this is Yogi Berra, who's a catcher for the New York Yankees, and he had all of these yogiisms that he used to say. And here's one that's really true. It's tough to make predictions, especially about the future. So what I'd like for you to think about is, how will we treat type 1 diabetes in 2022? How will we treat type 1 diabetes in 2025? And then finally, how will we treat type 1 diabetes in 10 years from now, in 2031? And at some point, will we ever be able to prevent type 1 diabetes in some of its forms? So I'd like to just end by discussing some considerations for all of the emerging and evolving therapies that will be talked about today. So one is, I want us to think about that not everyone who has type 1 diabetes is the same. So Sarah here developed type 1 diabetes when she was 10 years of age. Mr. H developed type 1 diabetes when he was 23 years of age. He then lived with type 1 diabetes for more than 50 years. Sarah lived with type 1 diabetes now for more than 10 years, is on a pump and a sensor. So how do we adapt? How do we choose therapies for these different individuals who have type 1 diabetes? And we have to consider the heterogeneity that I spoke about, the clinical heterogeneity, the metabolic heterogeneity, and the immunologic heterogeneity. Another thing I'd like for our group to consider as we talk about today are disparities in diabetes technology use as an example of this. So here's a study that was just published in January of this year that compared disparities in diabetes technology use in the United States compared to Europe. And there were great discrepancies. So as we think about new and emerging therapy, how do we make sure that the people who need these get these therapies, and that we address these current disparities? And then finally, I'd like to conclude that, what do you define as success in type 1 diabetes for therapy? Treatment is not a cure. And what is a cure? And this is something that I think that people would have disagreements about that. What if you had no maintenance therapy? You didn't need any injections or anything? That would be a cure. What if you didn't need to measure your blood glucose? What if you reverse beta cell autoimmunity through an immunomodulatory therapy? Would that be a success? So I think these are concepts that I suggest that we talk about in the discussion period of going on today. So I wanted to conclude there and thank you for allowing me to participate in this exciting session. This is my colleague, Marcella Brashova at Vanderbilt, where we direct this research group focused on islet cell biology. And we have a number of collaborators shown on this slide here. So I'll end there, and I look forward to having a discussion with you and answering questions. Thank you again for inviting me to speak.
Thank you very much, Dr. Powers. We now invite Dr. Melton to speak.
Well, thanks to the Endocrine Society, I'm glad to be joined by my colleague Al Powers and others, and excited to talk to you today about using stem cells to make pancreatic islets for diabetes. An important note here on the disclosures, I'll be describing some information from Vertex, which acquired a company that I started, and I serve as a consultant for Vertex now. So that's an important disclosure. As practically everyone on the Zoom today knows, type 1 diabetes has two main problems. There's the loss of beta cells, making the patients insulin dependent. And our idea there is to make more beta cells, and I'll talk about that. And secondly, there's an autoimmune attack on these same insulin-producing beta cells. And I'll finish up by talking about how we think about manipulating that immune attack to induce tolerance using stem cell-derived islets that have been genetically modified. I'll summarize some published work first to just say that some years ago, in fact, more than a decade ago, we set out to try to turn embryonic stem cells and pluripotent stem cells into functional beta cells. And by function, I mean cells that respond to glucose and secrete insulin in the right amounts. This shows a 6-step protocol we developed, which takes about 30 days. It involves about 15 different factors. And the important point on this slide is to look at the bottom left, where you see cadaveric organ donor islets, which have been stained for insulin. You see the green in the cytoplasm, and the transcription factor PDX1 red in the nucleus. And the stem cell derived islets that we're able to make called SC for stem cell beta cells on the right, you see a very similar, if not identical, pattern of insulin and PDX1 staining. Now, we've done enormous numbers of, I would say, transcriptional and proteomic kinds of tests on these cells to show how similar they are. But I thought I would just show one bit of information, which is a transmission electron micrograph. And what you can see here, as is well known, that a human beta cell packages its insulin into crystals, into vacuoles, shown here in the red box. And the stem cell-derived beta cells do the very same thing. And in my view, even though this is a kind of old-fashioned assay, the cell cannot make these crystallized insulin granules unless it is accurately turned on and off all the right genes during development in order to make this functional beta cell. We've published quite a bit on the genetic, as I say, molecular aspects of these cells. And today I want to concentrate a bit more on their function and what we might do next. So these cells, as I've said, do respond to glucose and secrete insulin. In the left panel here, you see three challenges with glucose: low and high, low and high, low and high, if you like, kind of like breakfast, lunch, and dinner. In each case, the cells secrete an appropriate amount of insulin following that glucose challenge. And on the far right are one of the best preparations we received of cadaveric islets, where again you see sequential insulin secretion following a glucose challenge. So the point here is that these cells are functional, and I might have just failed to mention that it took us more than a decade to figure out how to differentiate the cells into those functional cells. And I'm not going to say much more about that today. That's been widely published. What I am going to point to, though, is that we had shown that these cells work very well in several diabetic mouse models, in rats, and in monkeys. We had, actually, and also in pigs, with various colleagues. But given that I'm not a medical doctor or an endocrinologist, I work in a research lab here at the college, we weren't able to, of course, put these cells into patients. And that was done by this company, Vertex, I mentioned at the beginning. So let me just give you one, I think, exciting result that they've reported on. So they adapted and improved the protocol we devised to make these functional cells, did it with clinically compliant cells, scaled it up, went through the FDA for a phase 1/2 clinical trial. And as it happens, it was just earlier this week, on the 18th of October, or last week, where Vertex announced the results of this. I'll just quickly summarize them here. A patient was infused with these stem cell-derived islets. They call them VX, for Vertex, I guess, 880, using half the target dose along with immunosuppressive therapy, putting the cells into the portal vein. Now, importantly, this patient had been injecting insulin for 40 years. And in fact, in the year prior to the treatment, the patient had experienced 5 severe hypoglycemic episodes. And his insulin dose was 34 units per day, and there was no detectable C-peptide following mixed meal tolerance test. You see at the bottom in the table that Vertex provided that there was, as I said, undetectable evidence for insulin production. And following the infusion of these cells 90 days later, just 3 months later, there are therapeutically significant amounts of both fasting C-peptide and peak stimulated C-peptide. And the patient's hemoglobin A1C has already dropped just after 90 days. Most importantly, his required insulin dose dropped by more than 90%. As I've already said, I'm not a real doctor and I'm not accustomed to talking about patients, but I'm very excited about this result as a kind of validation of all the years of trying to make these functional human cells. They've now been tested, and I would suggest have met the test in a human patient. Now, of course, this is just one patient and it's only 90 days, so we shouldn't draw too many conclusions, but I think at this stage, it's sort of the best result we could have hoped for. I'm going to spend most of my talk now on kind of, I would say, planning for success. I'm going to assume that Vertex will continue this procedure and look ahead to how we might improve it, and then, as I've said, deal with the immune attack. As I noted, this patient is on immunosuppressants, as you would understand, because the cells derived from human embryonic stem cells, so it's an allo transplant. Now, let me correct one thing I've said so far, actually amplify it. Here's a picture of the stem cell-derived islets we make in our lab. And they make clusters that are the size of islets and are quite homogeneous. And an important thing you can see here is that if we judge the cells we want as beta cells, those express two factors, a transcription factor, NKX6.1, and C-peptide. And in a typical production or differentiation in my lab, we might get, as it shows here, 16% to about 30% of the beta cells. And what I had said so far implied that we make 100% beta cells. We don't. I'll show you in a minute what we do make. But now I want to move to the question of how would we get, what would I say, complete control over mastery of this differentiation process so that we could make like a designer islet. An endocrinologist could tell us exactly how many alpha and beta and delta cells they want. So here in kind of picture form, this is what it would look like. We want to think about the molecular and genetic controls that are used to make these islet cells. And as I've said, it's a 6-step procedure that we use where we add various growth factors and small molecules. And in the past few years, we have looked at all the chromatin changes that occur during that process. This was done by Juan Alvarez, where he looked at histone modifications, used ATAC-seq to look at open chromatin, and looked at DNA methylation. And the transcriptional changes were assessed by Adrian Veres, Aubrey Faust, and Nadav Sharom using single-cell sequencing. I might point out an interesting thing here is that this is the kind of information that one would never be able to get from human biopsies because this process occurs during human fetal development. So it's one of the advantages of growing the cells as we do at scale in vitro is that we can interrogate every step of this process. I'll show you one important result today that came from that. This is a TSNE plot of single cell sequencing of 37,000 some cells. And I want to make 2 points here. As many of you will know, you can show this data in different forms. I like this form because it looks like a lobster. And since I work in Boston, I think of it as the nicest way to kind of show the result. The cells we wanted are the purple cells. I'm trying to note here with my pointer. Those are stem cell-derived beta cells. Fortunately, we also make, with this protocol, stem cell-derived alpha cells that produce the counter-regulatory hormone glucagon. But we made these unwanted cells, enterochromaffin cells here in blue, and I'll say a bit about more of that later. And the other point shown here on the right is that once these cells have been made, they know who they are. They maintain their genetic programs over a 5-week period when they're just cultured in a simple media without any other inducing factors. So now moving ahead to the future, how would we get control of this process using genetic modification? And I show you here the general idea. There's a pancreatic progenitor that can make 2 of the cells we want, the beta cell and the alpha cell, but they also make this stem cell-derived enterochromaffin cell, which is not normally found in the islet. It's found in the intestine, and we don't want them, essentially. Yeah, we want to get rid of them. How could we do that? Well, there are many possible genetic perturbations one could use. You could do knockouts or CRISPR on-off screening, et cetera. And I'm going to tell you today just about genetic knockouts doing a whole genome CRISPR screen. But the hope here, is shown on the right, is to do a knockout, which, for example, will make more beta cells. That is, it will prevent alpha cell formation or enterochromaffin cell formation. Or you could also make fewer cells if you wanted and try to only make one type or another. This is a rather complicated procedure developed by Adrian Veres and Aubrey Faust, and I'm just going to summarize the results here today. What we do is we begin at the starting line with the embryonic stem cells, and then we want to know, can we make genetic modifications to tailor the differentiation process? It would restrict, if you like, close the door on making cells we don't want. And then I'll touch on the possibility that we could also make immune compatible cells, that is, cells which would not be rejected by the immune system. A schema of this system is shown here. We begin with about 300 million cells, usually, maybe 100 million. We infect them with a lentiviral library that has more than 4 guide RNAs for every gene in the human genome. The asterisk here indicates cells that have taken up the lentiviruses, which are provided at a low multiplicity of infection. We then select for cells that we only have those which have taken up the virus. And then using CRISPR-Cas9 and the guide RNAs, only one gene is knocked out at most in each cell. We seed them for differentiation, go through the 40-day differentiation, and then this color coding here is the purple are the beta cells we want, the red are the alpha cells we want, green are cells that we don't want, and blue are the enterochromaffin cells, another one we don't want. They dissociate the clusters, fix the cells with paraformaldehyde, stain for the markers, sort them, then undo the fixation. And the nice part here is you can just sequence for the guide RNA to tell you which gene was knocked out. It's not whole genome sequencing. It's just sequencing for the guide. And I'll show you one bit of data here. The data is presented in 2 ways. And I'm showing you a slide to kind of confirm that the experiment worked. If we knock out the gene PDX1, that's, as known to many people, required for beta cell development, and then ask how often are cells with that knockout becoming beta cells, you see that it's greatly reduced, statistically reduced. All of the knockouts here in the middle in this gray bar have no statistical significance, and each dot represents a single gene knockout. Another way to show the data is here on the right in this circle, which is what knockouts make a cell become a beta cell, an alpha cell, or an enterochromaffin cell. This says that if you knock out PDX1, you don't become a beta cell. This isn't a new finding, but it confirms that the whole genome screen worked. Similarly, you could see here for other known genes, I'll just point to ARX, known from the work of others to be required for alpha cells, and it's the opposite here. So as you might guess, we're focusing in on these genes here, which prevent the formation of enterochromaffin cells, and these genes up here, which make more beta cells. Since we don't yet have monoclonal versions of those cells, and we're actively doing that now, I hope the next time I speak with you all, I'll be able to tell you the results of those single genetic knockouts. I want to finish up by just pointing to the possibility now of using genetic modification to make what I might call, what should I call them, super cells, cells that have some kind of immune protection. They've either turned on or off a gene that prevents the immune system from rejecting them when they're transplanted. And my sort of last data slide here just shows the kinds of genes we're knocking in and out. We're looking at autoimmunity by stress resistance and alloreactivity by sort of obvious things, with clues coming from looking at the maternal-fetal interface and cancer cells. For example, PD-L1 and CD47 are well known to everybody. And then I described for you today this CRISPR screening. Last point is genetic modification the only way to do this? Again, pointing back to Sema Therapeutics and Vertex, Chris Thanos and his team have developed a device for encapsulation, which could also protect the cells from the immune system. And so if I were to try to look forward to the future as we were challenged here, I would say this is what I think is going to happen. And before I say that, I want to point out that almost every time I predict stuff, it doesn't come out that way. So this is what I'm thinking about. The first thing is to transplant these stem cell-derived islets into patients with immunosuppression. And I was pleased today to be able to share with you Vertex's exciting results with the first patient. The next thing that I think will happen, and I hope that'll happen next year, would be to try the encapsulation device to transplant into patients. That device is very cleverly designed because it keeps cells within 150 microns of blood vessels and oxygen. And in pigs, it's done very well. So now we'll see how it does in humans. Then thinking ahead longer term to the future, could we genetically modify the cells to make the right composition we want and protect them from the immune system? So the dream here would be when a family is shocked by the diagnosis of a type 1 diabetes in their children and they go into their doctor, the endocrinologist says, well, don't worry, I have some of these cells in the freezer. I'm going to inject them into you and they'll make up for your lost beta cells. And if you need to come back in a year or two, she'll go into her freezer and get another batch and inject those into you. In my opinion, we're now in a position where we can seriously think about changing the life of diabetics who have to monitor their blood sugar, inject insulin, care for their diet and exercise so attentively every day by providing them with, I dare say, sort of nature's solution to controlling diabetes. In my last slide, I thank the people who've done this work with me, and I'd particularly like to note here Adrian Vera is at the top. I won't unfortunately be able to join you live on the day of the presentation, and Adrian, who's done much of this work, will stand in for me. Thank you again for the invitation, and I hope you're all enjoying the meeting.
Thank you so much, Dr. Melton. We will now open up the Q&A with Dr. Powers and Dr. Adrian Veras from Dr. Melton's lab, who's filling in for him today. Please type your questions into the chat box on the right side of your screen. I'm going to turn it over to my colleague Jenny now to ask the questions.
Thank you, Colleen. I'll go ahead and start with a question we received for Dr. Veras. Dr. Veras, can you tell us, does cell therapy really represent a true hope or just a fantasy for treating diabetes?
I think it's always been a dream to achieve this. I think there's reasons to think it's not just a fantasy. To use the language of the question is twofold. First of all, it's already a reality when we look at deceased organ islet transplantation. This is a therapy that is in use in some countries around the world. It's immensely limited by the need for immunosuppression, as well as the limited availability of islets for transplantation. And I should also say now, especially with this first patient's results that we've seen from Vertex, and I should also add I have no affiliation with Vertex whatsoever. I think we're one big step closer to this being a true hope. And I think combining those two, there's a matter of engineering and being able to develop those cells. But I think it's going to be a matter of removing barriers so that even for a relatively uncomplicated person with type 1 diabetes, this is an option that's on the table that can be deployed when appropriate.
Great, thank you. Question for Dr. Powers. Is there data about beta cell mass in young type 2 patients? We're talking about onset of ages 12 to 16 and not MODY. Is this different from older onset type 2?
Yeah, so this is a great question about beta cell mass. And so it's a very difficult question where we don't really have the data that we need. We know that beta cell mass measured at autopsy, we can find that it varies 3 or 4-fold over a population. And there are a number of studies looking at beta cell mass in individuals who died with type 1 or type 2 diabetes. What we don't know is the longitudinal nature of the beta cell mass. We don't know that those individuals, let's say, who are in the lower quartile of beta cell mass, were they always in that quartile, or has there been a change in beta cell mass? So we don't really know whether it goes in humans, whether it goes up in pregnancy, up with obesity, down with the development of type 2 diabetes. And that's because we don't have a way to non-invasively measure beta cell mass. So it remains an unknown about what beta cell mass is in young individuals with type 2 diabetes. Thank you.
Thank you, Dr. Powers. Dr. Veras, I have another question for you from our Q&A. Super exciting innovation by Vertex. Is this expected to be a one-time injection?
I think that's a great question as well. We don't know yet. I think it depends on the performance of the stem cell product itself. Again, looking at lessons that we know from deceased organ donor islet transplantation, there are cases where the first infusion provides some function, but not sufficient function to not sufficient function for the patient in question, and a second infusion is required. So that there's already precedent for going a sort of step-by-step approach. Then another format of this question is kind of what is the durability of the cells, both in terms of their own ability to function as well as their interaction with the immune system. And there, I think both paths are possible. One is if we are able to create if a product can be created that can last for 5 years at a time and needs to be replaced on the span of several years, then that might be one path where there's a certain expected rhythm of repeating the injection. That said, on the other hand, I think the goal that we're all aiming for is the advantage of cell therapy is this potential for a one-time cure. And that's a place that, if it's limited by engineering, then continued research efforts and research will continue to aim towards that goal of minimizing and being able to do a one-time injection.
Thank you, Dr. Veras. Let's see, I have a question for Dr. Powers. Can serial C-peptide levels be used as a surrogate measure for beta cell mass?
So C-peptide, as we know, is a product of the beta cell, just like insulin, but because of its longer half-life, it's able to reflect better endogenous insulin secretion. So the answer to that would be yes, meaning the individuals who have type 1 diabetes or who have relatively insulin deficient type 2 diabetes will have lower C-peptide levels. Thank you.
Thank you. I have a question for Dr. Veras. Is this a role eventually for adult stem cells to be used rather than embryonic stem cells? This might be ethically more appealing for some.
So in terms of the actual differentiation process itself, whether we're using induced pluripotent stem cells, which are typically created by reprogramming adult-derived cells, or embryonic stem cells, the protocols being developed for both should be compatible or adaptable for either source material. I think on the question, there's a broader question here, which is, do we have to start with a pluripotent stem cells, whether it's induced or embryonic? And as far as we know, this is actually a relatively kind of hotly debated question in the field of, are there resident stem cells in the pancreas that can be used to generate those beta cells? And the answer, generally speaking, in the field there is no. The cells that are present in the pancreas have very limited ability to create new beta cells. So we're not in a potential where we could harvest a patient's own pancreatic stem cells and sort of use those to do the reprogramming. But in terms of ethical considerations having to do with embryonic stem cell use, I think the existence and ability to use iPSC-derived beta cells should be able to resolve that in the future as well.
Thank you, Dr. Veras. Dr. Powers, I have another question for you. Given the heterogeneity of type 1 diabetes and the disparities that exist in treatments, what are your thoughts on how we can address some of the disparities that exist with current state and future management?
So I think there are two parts that this questioner brings up. One are the disparities, meaning that individuals who have insulin deficient diabetes have access to the latest types of insulin, the latest technology. And so I think that's something where, especially in the United States, that we have ongoing concerns with that. The study I mentioned in my talk about the disparities around technology between Europe and the US was particularly striking. So this is something that we need to address as society, and that'll require insurers and the government and patients to be involved. The other part of the question was the heterogeneity, about that everyone who has type 1 diabetes is not the same. And so there, I think we're going to need individualized or approaches that might be customized to a person. So for example, someone who is very insulin deficient, a type of transplant of a cell might be very attractive, because even if it provided just some insulin, that's likely to greatly improve the glycemic control. And whereas other individuals who are very young, there may be things where one thinks of some type of immunomodulatory therapy that might be more focused on. So I think that as we consider the heterogeneity of what it means to have type 1 diabetes, we're going to have some approaches that are better for one type of patient and other approaches that will be for another type of patient. Thank you.
Thank you. We have a question about nutrition and lifestyle. If you want to take this one, Dr. Powers. Nutrition blogger Bea Paredes is asking, do you think the nutritionist should work hand in hand with physicians? What about nutrition and lifestyle? How does that fit in with these approaches?
Yeah, certainly. I think the foundation of treatment of all forms of diabetes is appropriate nutrition. And there are a variety of changes that people are incorporating in their diets, such as the amount of carbohydrate in the diet, low carbohydrate diets, especially in type 2 diabetes. These have shown great potential. So I think no matter how a person's diabetes is treated, whether it's by a pump with a sensor or a stem cell transplant or regenerated beta cells from an endogenous source, nutrition is going to be, I think, important in matching those insulin glucose uses. Thank you.
Thank you. Dr. Veras, if you're successful in protecting stem cell-derived islets from immune rejection, isn't there a danger of new mutations arising after transplantation that led to the tumor formation? And if so, what if the patient's immune system cannot reject these newly formed tumors?
Absolutely. So that's a great question. Is the immune system, obviously in a situation of autoimmune disease, is causing harm, but the immune system also allows for appropriate regulation of endogenous cells and tissues. And as we're manipulating the reduction of harm, is there a way to apply this trade-off? I think there's a number of different elements there that help in answering this question. One is specifically for the case of the beta cell and the islet, we're really talking about essentially post-mitotic cells. So these are cells that don't divide anymore. That's true of in kind of natural endogenous beta cells, where the rate of division is very small over time in most contexts. And that's also true in the product of stem cell derived beta cells. In that lower rate of division really provides one sort of layer of how difficult the problem of regulation is. The next layer is, it's important to know that in parallel, as different labs are working on modifications that allow for immune protection, there's a lot of activity in the stem cell, in the regenerative biology field writ large, not just in the beta cell context, of identifying ways where we can introduce safety switches. So you could imagine, for example, cells that have various mechanisms that are responsible to, in the presence of a small molecule, would then be able to shut down the grafted tissue. And this question of how can we engineer these safety switches, especially once we're removing the endogenous immune response, for example, are very important questions that the field is working on currently.
Thank you. Dr. Powers, I've got a few more questions for you. Do you have an opinion about the keto diet or other very low carb diets with respect to helping manage type 1 diabetes?
Yeah, so this is a complex question, and so I think initially there was great hesitance to apply low carb diets in type 1 diabetes, and I still think that's sort of the prevailing view, that it has to be done very cautiously. There are some individuals, though, who are advocating low carbohydrate diets for individuals with type 1 diabetes, as I mentioned, has been used in type 2 diabetes. So I think that should only be done in close consultation with the individual's physician.
Thank you. Dr. Veras, I think this one's for you. Hypoglycemia is a huge problem when treating diabetes. Is there any work on the function of alpha cells or the need to transplant alpha cells when transplanting beta cells?
Absolutely. So this is a great question and an interesting answer to this, which is, oddly enough, alpha cells we succeeded in making alpha cells earlier than making beta cells, insofar as a lot of early protocols on the road to existing beta cell differentiation protocol make a mixture of cells, and part of the cells in that mixture are those alpha cells. We see, in fact, that when we actually try to make the beta cells in the differentiation, we make typically about a matching number of alpha cells. Now, we don't yet know why we do that. We are, it's actually a very interesting developmental biology question that we're actively studying in the lab. But as it is today, when we form these stem cell derived islets, they contain a significant fraction of alpha cells that should be able to function and work together with the beta cells for appropriate regulation of insulin and glucagon secretion.
Thank you. Does exogenous insulin suppress remaining endogenous insulin? Could that lower C-peptides in type 1 diabetes?
Yeah, maybe I'll take that one. So the question is, does exogenous insulin lower or maybe prevent endogenous insulin production, or even prevent the regeneration of beta cells because you wouldn't have the hyperglycemic stimulus? We don't know, but I think in in animal models, one would suggest that the regeneration would be stimulated a bit by hyperglycemia and would not be suppressed by exogenous insulin replacement.
Thank you, Dr. Powers. Are there any molecules that have been shown to be able to regenerate at least partially the endogenous beta cells in type 1 patients?
Well, so when one moves into the human arena, there are two challenges. One, what molecules you deliver, and two, how do you deliver them? Because many of the molecules that are delivered would stimulate growth of many different types of cells. So you'd like to either have a molecule that only worked in beta cells. I think that's unlikely. Or you'd like to be able to deliver a molecule that stimulated regeneration and proliferation, and you could restrict or limit the delivery of that molecule to the beta cells. And so that's still a work in progress. There are some molecules that stimulate regeneration. What's currently lacking is the ability to target and deliver things specifically to the human islet or to the human beta cell. Thank you.
Next question. What is the role of dedifferentiation and redifferentiation of beta cells and transdifferentiation of other existing cells in the pancreas to beta cells, depending on the levels of blood glucose?
Adrian, maybe I'll let you take that first, and then I'll add a comment.
Absolutely. I think this question of how cells transform from one cell type to another, first of all, in general, like, do they have this ability, is hotly debated in our field. There's some form of evidence that in some cases alpha cells may be able to transdifferentiate into beta cells, and then the question is, of course, can we enhance that process? That said, this is not necessarily a prevailing view that this is in fact possible. In terms of dedifferentiation, where beta cells lose their phenotype and of a beta cell in particularly situations like type 2, there's quite good evidence to this. We can even see like loss of insulin expression in cells that otherwise should be beta cells. That said, the ability for those differentiated cells to do something useful, to either redifferentiate into one cell type or another, again, is an area of relatively little evidence and relatively little kind of consistent and convincing evidence. And again, this is a situation where the challenge of how these things might occur in humans versus how they occur in the various animal models we have at our disposal is also, like, translating across these different models is also proving to be quite challenging.
Yeah, I think there are two points that I would add to that. One is what Adrian says about the dedifferentiation in type 2 diabetes. If those cells could be redifferentiated, that would be endogenous cells that are there. Now, I will say that the clarity of that dedifferentiation occurs in human type 2 diabetes is still the evidence for that is limited. It does occur in rodent models. So I think that's that. The second is that when we talk about changing cell type fate, while attractive, there are also many of the issues that Adrian spoke about in terms of safety and how do you guide that process. I mean, those differentiation things that happen in vivo are much more difficult to control than when Adrian does that in vitro, where the cells can be differentiated to a certain state and then introduced into a person.
Great. Next question, is there any use for insulin like growth factors in diabetes?
Yeah, I mean, those obviously can make cells grow, and they are produced by the pancreas. And so right now, I don't know of a clear role for them in the regeneration of human islets in vivo. I don't know, Adrian, if you want to add anything to that.
They're not a core factor that is used in the process of making the cells in vitro as well. They're a common growth factor for their ability to activate certain signaling pathways in vitro. But in terms of if we had to list the sort of top 10 most important signaling pathways that we use in the generation of beta cells, IGF-like molecules are not in that list.
Thank you. How do you go about controlling the growth of transplanted islet cells so as not to reach the level of insulinoma?
So that's a great question. The short answer is that the cells do it for us already, in the sense that endocrine cells in the islet are by and at large post-mitotic. If we look at a typical islet section, we'd expect about 1 cell in 1,000 to be dividing. This can change in certain contexts, most notably pregnancy, where that number can go up to about 1 in 100. But by and large, the cells are post-mitotic. That's true for the endogenous endocrine cells, and is also true for the lab-made in vitro derived cells. And we actually see that there's sort of a clear mechanistic linking here, which is as a cell who is a pancreatic progenitor expresses the right set of genes to undergo the transition to an endocrine fate, that transition is also associated with pausing the cell cycle. So, we've seen those cells be post-mitotic, and we've also tested the post-mitotic natures of the beta cells themselves. Like we know that when we transplant them, their rates of replication match roughly what we expect of endogenous cells. Now, there's always this question of purity in terms of when we put in beta cells, if there's remaining cells that are capable of dividing because they're a pancreatic progenitor or even an earlier progenitor in the process, what do we do about that? And that's a question broadly for the field of cell therapeutics, of how we're actually able to develop the appropriate clinical grade manufacturing and quality control practices to be able to really rule this out in particular batches of cells that are going to be used for human transplantation. And that's an area of very active research where animal models, very novel forms of cell type profilings that are available today are really helping us answer those questions.
Thank you. On that related note, I have a question. Islet mass has been reported to increase during pregnancy. Is this still believed to be true? And if so, what is known about regulation of this process?
Well, it's certainly true in rodents where the islet mass goes up and it goes back down after delivery. In humans, there have been very few studies of the human pancreas in women who unfortunately died during pregnancy. But it appears from the limited studies that the large increase in beta cell mass that occurs in rodents during pregnancy does not occur in humans. There's clearly increased beta cell function and increased insulin secretion. And so my view is that that's what happens in a human pregnancy, more increase in function, but a modest or rather limited increase in beta cell mass.
Has the function of pancreatic polypeptides been elucidated?
The hormone, I don't think I know of a clear function from pancreatic polypeptide in humans. I don't know, Adrian, you want to comment?
I was hoping Dr. Powers would tell me what it does finally, but I don't know of a function myself either.
Fair enough. We're able to find out more. We'll get back to you. The next question, are there in vitro models that would benefit the field? I hear about microfluidic stuff, but don't know where it could be helpful.
Adrian, I'll let you take that because I know you've studied some of your cells in some such devices.
Absolutely. So the answer is a mix of yes and no. One is, as we're thinking about studying the function of these cells and how they're behaving, the more we can control the local environment, for example, to simulate a graft, for example, to simulate an interaction with the immune system, the more systems like microfluidics, which allow us to have these very delicate channels of very precise size and really give us exquisite control over the local environment of an islet, really come into play. And this is the area that they shine. So they're excellent tools for study in that regard. That said, one of the big challenges of kind of the field that really the field has essentially solved, having to do with making sufficient in vitro beta cells to cure a person, which requires essentially several billion cells. That's a scale of tissue culture that is quite unusual for the field in general. In the beta cell community, we solve this by growing cells in these 3D organoids that are sort of grown in large scale 3D culture. That's a setting where unfortunately, systems like microfluidics or systems that really allow you to control the environment of one cluster typically fail once you're trying to control the environment of millions of clusters floating together. So there's some value in that sense. One place where we're actually, these two systems interact is if you're trying to take all those hundreds of thousands of stem cell derived islets and coat them in a particular immunomodulatory environment, that's a place where now microfluidics comes back and allows you to do that coating in a efficient and very controllable way. But typically there's this challenge of, if you're trying to work at a clinically relevant scale, which we've done for some time in our lab now, that's already a kind of an imposition on the type of science we can do, and that local control is less doable.
Thank you. One more question. Are cells in nesidioblastosis or beta cell hyperplasia autonomous or simply genetically turned on? Can this be used to increase beta cell mass?
Adrian, you want me to go
Yes.
first? Yes. So nesidioblastosis, which is seen primarily in the pediatric population, it can either be focal or diffuse growth of beta cells, and there are several reasons, some of which are genetic for that. People have tried to learn from that to see if that could be used to promote the growth of normal beta cells. And as of now, there's not been a clear path for using that in a way that you could have controlled expansion of normal beta cells. But a great idea.
I think on one thing I want to add to this is these types of rare disorders that we can learn from are crucial to our field, in part because the beta cell is a very special cell in the sense that it's present in islets and only in the pancreas. If we actually look at the rest of endocrine cells, even the alpha cells, the delta cells, and we actually look at where they're distributed in the rest of endodermal tissues, like throughout the gut tube, we see substantial, like the cells are more spread out throughout the gut tube, and very often there's many variants of the same cell type in many different locations along the gut tube organs themselves. So things that allow us to understand why is it that beta cells form only in essentially one place and only in one context, which is the context of the islet, can give us clues to really understand and even loop this information back into better controlling the process of beta cell formation itself. So we're finally able to do it, and we've been able to do it for a few years, but there's still kind of enormous potential left in how much we could simplify, streamline, and optimize that process.
Wonderful. Well, thank you to you both for answering all those questions, and to the audience for such thoughtful questions. It's been a great discussion. And I'll now turn it back over to Colleen.
Great. Thank you so much, Jenny. Yes, that was great participation and some really awesome questions. We really appreciate it. We're going to take about a 10-minute break and regroup for our next session around 12:20 Eastern. Welcome back, everyone. We're pleased to introduce the speakers of our second session on advances in pharmacotherapy. Today we have with us Dr. Elizabeth Sequist of the University of Minnesota, Dr. Juan Pablo Frias of the National Research Institute, and Dr. Yogesh Kudva of Mayo Clinic. Dr. Sequist, we invite you to please speak first.
Well, thank you very much. And I'd really like to thank the Endocrine Society for inviting me to be part of this really exciting symposium. It's such a joy to celebrate 100 years of insulin. I'm really feel honored to be part of the symposium. My task for the next few minutes is to talk about the next generation glucagons that are available for severe hypoglycemia. I think all of us are aware that hypoglycemia is a major problem for patients with diabetes. It is the factor that limits the management of the disease. The fear of hypoglycemia prevents patients from obtaining the reduction in microvascular risk that is associated with good glycemic control. And the impact of hypoglycemia can range from mere inconvenience, which I think is always more than inconvenient, to coma, seizures, and even death. We also know that recurrent hypoglycemia leads to impaired awareness of hypoglycemia, something that can be very, very devastating to patients. With recurrent hypoglycemia, there is a shift in the glucose threshold that elicits the counterregulatory response. That is, it takes a lower and lower blood glucose to elicit that response. There's also a reduction in the magnitude of the counterregulatory hormone response. And for patients, the first symptom of hypoglycemia can be neuroglycopenia. This is estimated to occur in about a quarter of patients with type 1 diabetes, and patients who have impaired awareness have an increased risk of severe hypoglycemia by more than 3 to 6-fold. So this is a common problem in our patient population and something we really need to think carefully about. We've long known there's an undeniable need for glucagon. We know when we're prescribing insulin, we should think about prescribing glucagon, and anyone who's at increased risk for level 2 or 3 hypoglycemia, that is glucose values less than 54 or severe hypoglycemia, should be prescribed this so it's available should it be needed. In addition, we need to be sure that the caregivers, school personnel, family members, whoever is going to be around these individuals should know where the glucagon is, when to use it, and how to administer it. And we know that we should instruct people that glucagon should be used to treat hypoglycemia when someone is unconscious, when they can't take oral carbohydrates by mouth successfully. Now, in the past, we have had glucagon kits. There's 2 different manufacturers. They're both shown here. And this slide has a lot of elements on it, and it really highlights to me the difficulty the people have had with using these products. If you look at it, glucagon, traditional glucagon comes in this kit, and in the kit there is a pre-filled syringe that has saline in it. There is a freeze-dried glucagon in a vial, and someone needs to take the cap off the needle, put the diluent into the vial with the glucagon, make sure it's mixed up, draw it back up, and then give it to their loved one who is unconscious on the floor. This is very intimidating to many family members, and it often results in them never using the glucagon. And so that means our patients are hypoglycemic, seriously so, with unconscious for longer than they need to be. Fortunately, in the last couple of years, we've had new glucagon products come on the market that may enhance, that are probably easier for our family members to use. One of these products is a nasal glucagon product shown here. It comes in this yellow holder, and you flip off the top, and there is the device right there. You can see there is a nasal, at the top of it is something you can put in the nose. This is completely nasal-free, needle-free. It's a dry powder, and when a person pushes the bottom of the canister towards the top, it releases the powder into the person's nose. It's ready to use. It doesn't require any reconstitution. It's very compact, portable. It's a single use. It's all contained. No one needs to put anything together. The person with hypoglycemia does not need to inhale or breathe deeply. It's automatically dispersed into their nose. It has a shelf life of 2 years and it's stable up to 30 degrees. And the dose, a single 3 milligram dose, has been approved for people 4 years of age and older. There also are new liquid stable native glucagon products. Here is one of them. This manufacturer creates pre-mixed ready-to-use forms. There's 2 of them. There's a pre-filled syringe and an auto injector. One of the pens has no visible needle. This too has a long shelf life, 2 years, and can be stored at room temperature. And there are doses for pediatric patients, for just less than 12, as well as adults and adolescents. Another liquid glucagon analog is made by a different manufacturer. It also has pre-filled syringes and auto injectors. The auto injector has no visible needle. It too can be stored at room temperature, or it can be refrigerated. And this product has a shelf life of 12 months if stored at room temperature. And a single dose is approved for patients over the age of 6. Now, these products seem to work very well. I'm going to share some of the experimental data that tells you a little bit about how they compare to the emergency glucagon kit. And this is data taken from a paper that Mike Rickles and his group published a couple of years ago, where they gave adults with type 1 diabetes insulin. They made them hypoglycemic down to a level of 50. That's shown in A, the A graph here. And we see that time 0, the nasal glucagon product is administered, and we can see the time to recovery. So in the black line are patients who were given nasal glucagon. In the dotted line are same patients, randomized order, given the intramuscular glucagon product. And we see that the lines overlap. They're the time to recovery, the time between a glucose value of 50 when the glucagon products were administered to when their blood sugar is in the euglycemic range is approximately the same. In panel B, we look at the plasma glucagon concentrations that can be measured in the patients following administration of the glucagon product here again in black versus the intramuscular glucagon or subcutaneous glucagon shown the emergency kit in dotted line. And you see they achieve very similar glucagon concentrations. The study also looked at the time to achieving recovery and then side effects. So if we first looked at the graph, we can see that again, solid line is the intranasal administration at time 0 during hypoglycemia. The intramuscular is given in the dotted line. And we can see that by 20 minutes, 90% of the patients had achieved euglycemia following intranasal glucagon administration, and 100% of the patients had given the intramuscular. And if we look at by 40 minutes, all the patients who had been administered these products were to euglycemia. So very similar time course of recovery from hypoglycemia between the nasal glucagon and the typical emergency kit. Table B shows the adverse events that people have in response to these treatments. And not surprisingly, it's glucagon, and people have abdominal discomfort, nausea, and vomiting. Really no difference between the 2 products, the intranasal or the intramuscular. There are some more nasal-related side effects related to the administration of intranasal glucagon. You can see nasal congestion, nasal discomfort, that sort of thing, and some more headaches, ear pain, some more things probably related to the product itself and the way it's administered. But the patients generally tolerate these very well, and is not a barrier to using the product again in the future. I also want to share a study that we were able to participate in that looked at how well caregivers were able to give this nasal product in real life. What we did was recruit adults with type 1 and their caregivers. They had to come into the clinic, and we instructed them in the use of the 3 milligram nasal glucagon product. The product was sent home with the 2 of them, the caregiver and the type 1 patient. And the caregiver was instructed to use it in the event of severe hypoglycemia. These were all patients who had a risk of severe hypoglycemia based on their previous history. 69 patients with type 1 diabetes had an episode of moderate to severe hypoglycemia during the period of follow-up, and 69 of these patients were treated with nasal glucagon. 95% of the patients given the glucagon product recovered within 30 minutes. Here are data collected from the caregivers that help us understand how they feel about using this product. So we asked them, tell us about the time taken to administer nasal glucagon. And we see that the vast majority of them gave it in less than 30 seconds. We look at understanding the kit instruction. Was it easy to understand? And again, we see people thought it was very easy to understand. Was it easy to administer? People thought it was very easy to administer. And overall, people were very satisfied with this. And this compares to literature using the emergency kit where many patients, many caregivers never figure out how to actually administer in a timely fashion. So just to conclude, new formulations of glucagon that are easier to use and store than the emergency kit have become available. Efficacy and adverse events are similar to the IM glucagon. Intranasal glucagon may cause some nasal irritation, and clinicians should prescribe glucagon to all patients at risk for hypoglycemia. Thank you for your attention.
Thank you so much, Dr. Sequist, for that great presentation. We now invite Dr. Frias to speak.
Thank you very much for this kind invitation to speak about once-weekly insulins, which are in development for the management of type 2 diabetes. And as you'll see, what the studies to date have shown is that they're equally effective for the treatment of type 2 diabetes versus the second-generation basal insulin analogs. So by way of background, we know that type 2 diabetes is a very complex disease with defects in various organs and tissues which contribute to hyperglycemia. And we're very fortunate today to have a number of different classes of agents, in addition to insulin, to help reduce glucose in patients with type 2 diabetes. And one of the key defects in type 2 diabetes is a progressive decline in beta cell function. Therefore, most patients, if not almost all patients, will require insulin therapy at some point in the natural history of their disease to both attain and maintain good glycemic control. So if we look at the key tenets or principles in the management of type 2 diabetes, we should aim to achieve the best possible glycemic control with the least possible side effects, in particular hypoglycemia and weight gain. And this often requires combining medications with different mechanisms and complementary mechanisms of action. We should also use agents that help reduce morbidity and mortality from atherosclerotic cardiovascular disease, heart failure, and chronic kidney disease. And very importantly, as I mentioned, based on this progressive decline in beta cell function, many patients, if not most, will require insulin therapy during the course of their disease. And if we look at guidelines, and this is from ADA and EASD, with respect to injectable therapy in patients with type 2 diabetes, insulin should be at least considered as the first injectable therapy, particularly in patients who are very poorly controlled on oral agents. And certainly if we start with the GLP-1 receptor agonist and the patient is still not achieving control, adding basal insulin should be considered at that point. And this actually can be done in the form of a fixed ratio combination of basal insulin and a GLP-1 receptor agonist. And that's pertinent to this discussion because, as we'll see, the once weekly basal insulins in development can be or potentially will be formulated with GLP-1 receptor agonists as well. And there are many unmet needs today with respect to basal insulin therapy. One key issue is therapeutic inertia, or taking a very long time to initiate basal insulin therapy in patients despite their very poor glycemic control. And this is due oftentimes to myths about insulin therapy, that insulin may cause complications, reluctance by patients to initiate insulin, and oftentimes by clinicians to start insulin therapy, injection burden and complexity, stigma associated with diabetes, and actually people watching, perhaps giving their insulin injections. And all of these can lead to challenges not only in initiating insulin therapy, but also with adherence and persistence with therapy. If we look specifically at basal insulins, there's been a very nice evolution over time since the discovery of insulin in 1921. Protamine to enhance or prolong the time action profile, initially of animal-based insulins, but then in 82 of recombinant human insulin. And very important, the first basal insulin analog, insulin glargine U100 in 2000, the second so-called first generation insulin detamir, and more recently, the second generation basal insulin analogs with prolonged activity, flatter time action profiles, insulin degludec and insulin glargine U300. And this really is a very important study, the Treat the Target trial published by Matt Riddle and Julio Rosenstock in 2003, which looked at the first generation basal insulin analog, insulin glargine U100 compared to MPH, showing that we can get to the same hemoglobin A1C with these analogs, but with less hypoglycemia, and in some cases with less weight gain, likely because of the lower risk of hypoglycemia. And this is also what's been seen as we've gone from the first generation basal insulin analogs to the second generation. We see here insulin glargine U300 compared to insulin glargine U100, the time action profile, longer duration, a flatter profile that you see on the left in green, this leading to similar hemoglobin A1C change, but with lower risk of hypoglycemia. And likewise with insulin degludec, lower, a flatter profile, longer duration, less variability day to day compared to insulin glargine, again leading to less hypoglycemia despite similar A1C reductions. And we've also seen with insulin degludec, even the very long duration of action, studies that have looked at more flexible dosing as well. And though this is not in the label to dose 3 times a week, certainly we have flexibility during the day, again, due to this prolonged action of the second generation basal insulin analog, which brings us to the weekly basal insulin analogs that are currently under development. So there are 2. One is Eli Lilly's basal insulin FC, which is referred to as BIF. And this is a fusion protein, and it combines a single chain variant of insulin with a human IgG FC domain. So this is a method by which its action is prolonged, its half-life is prolonged, and it can be given once weekly. And this is the same mechanism that's used by many different drugs, including the GLP-1 receptor agonist dulaglutide, and BIF is currently in phase 2 of clinical development. And Novo Nordisk has a once weekly basal insulin analog called insulin icodec, and this binds very strongly to albumin, and that is the way by which its half-life is prolonged, and it has a pharmacokinetic profile allowing for once weekly administration. It has completed phase 2, and I'll go over these trials, and is currently in a very robust phase 3 program called the Onward program. And the advantages, as you can imagine, of having a once weekly basal insulin is greater convenience for the patient, which could lead to improved adherence and persistence, reduced treatment burden for patients and caretakers. So you're talking now about potentially 52 injections annually as opposed to 365 injections, improve healthcare-related quality of life, patient-reported outcomes, and also the ability to formulate with once-weekly GLP-1 receptor agonists. Again, the potential down the line for a fixed ratio combination of a once-weekly basal insulin and a once-weekly GLP-1 receptor agonist. So let's review the 2 once-weekly basal insulins that are currently in clinical development. Again, BIF, fusion protein of a single chain insulin variant combined with human IgG FC domain. It's very selective for the insulin receptor compared to IGF-1, so low mitogenicity potential. The pharmacokinetic profile, again, is consistent or allows once-weekly subcutaneous dosing. It's formulated so it could be used in a single-use or multi-use device. And again, importantly, it can be co-formulated with weekly incretins and has a low immunogenicity risk. This is a phase 2 study which has been completed. I actually presented this at the Endocrine Society meetings last year and also at EASD this year. It was a phase 2 study in patients with type 2 diabetes who were already on basal insulin. The mean A1C of baseline was 8.1, and we studied over a 32-week period 2 different dosing algorithms of BIF. So this was administered once weekly, one with a fasting glucose target of 140 milligrams per deciliter, the second arm with a target of 120 milligrams per deciliter, versus insulin degludec, typical treat to target algorithm with a fasting glucose target of less than 100 milligrams per deciliter. And the glucose targets were higher in BIF for safety reasons, as this was the first time that this medication had been studied in an outpatient setting. Dose adjustments were made based on fasting glucose. And the BIF doses, the adjustments were made in milligram increments in this study. And here we see the primary endpoint. In the bottom lines, you see a nice reduction in hemoglobin A1C over the 32 weeks. You see the absolute change, mean changes on the right. And these were non-inferior. So there was non-inferiority between the once weekly BIF, both arms, and insulin degludec. But importantly, there was less hypoglycemia. And this is looking at hypoglycemia less than 70 milligrams per deciliter, both documented hypoglycemia throughout the day and nocturnal hypoglycemia. The event rates were lower with BIF compared to insulin degludec. So to summarize, there was similar glycemic control despite the fact that there were higher glucose targets, fasting glucose targets with BIF compared to insulin degludec, significantly lower rates of documented hypoglycemia with BIF. Now we have to take into consideration that the target for degludec was lower than for BIF, and BIF had an overall similar safety profile compared to insulin degludec. And there is a large phase 2 program that's currently ongoing in type 2 diabetes and type 1 diabetes, where comparing again to insulin degludec, but with all arms having fasting glucose targets that are similar and less than 100 milligrams per deciliter. If we now turn to icodec, so this is a novel basal insulin analog that strongly but reversibly binds to albumin, and that's the mechanism by which it prolongs its action. It has 3 amino acid substitutions, which reduces enzymatic degradation. And then the C20 fatty diacid moiety, which binds to albumin and also reduces receptor mediated clearance. There have been 3 phase 3 trials that have been completed and recently published. They all compare insulin icodec to insulin glargine U100. 2 of these trials in patients who are naive to insulin, and 1 trial in patients who are already using basal insulin. And in these studies, as you'll see, loading doses as well as titration strategies were assessed versus insulin glargine U100. So we'll start with the paper that was recently published actually last year by Julio Rosenstock in the New England Journal. This was a study in patients who were insulin naive. They were on metformin with or without a DPP-4 inhibitor, and initiated weekly insulin icodec versus daily insulin glargine. Importantly, this was a placebo-controlled trial. And also it's important to note that the starting dose for insulin glargine was 10 units, sort of a very typical starting dose, and that was multiplied by 7, so 70 units once a week for icodec. And the glucose targets that were being aimed for with titrations was 70 to 108 milligrams per deciliter. And you can see the dose increments there, 0, 2, or 4 units for insulin glargine, and basically just multiplying that by 7, 0, 14, or 28 units increments, depending on the fasting glucose for icodec. And the primary endpoint was a change in hemoglobin A1C, and you see very nice reductions in A1C to a mean of less than 7% in both study arms, and numerically better A1C reduction with icodec. The estimated treatment difference was about 0.2%, which was not statistically significant, but also very nice target attainment with approximately 72 and 68% of patients achieving an A1C of less than 7% with icodec and insulin glargine, but numerically again higher with icodec. There was less insulin use over the 26 weeks with insulin icodec compared to insulin glargine. You can look at the approximate daily doses of insulin glargine at the end of the study. approximately 40 units compared to 33 units with Icodec, and similar increase in body weight as might be expected with both insulin formulations. Importantly, there was more level 1, so hypoglycemia between 54 and 70 milligrams per deciliter, so mild hypoglycemia with insulin degludec compared to insulin glargine, but more clinically relevant hypoglycemia, so level 2 and level 3, was fairly comparable between the two insulins. And the authors basically stated that perhaps this target of 70 to 108 was a little too low, and this could have prevented some of the level 1 and obviously the level 2 and 3 hypoglycemia as well. There were 2 additional open label phase 2 trials that have been published recently. One, and these are 16-week open label studies. One looked specifically at patients already on basal insulin and was addressing the question of whether a loading dose was needed as the first dose of once weekly basal insulin. And the second trial looked at patients who were insulin naive, also 16 weeks, and looked at different dose titration algorithms. So we'll start with a trial looking at the loading dose or no loading dose. And basically in this study, patients were already on basal insulin, and they were then switched either to continue that dose if they went to the insulin glargine arm. That dose times 7 was given if they went to the no loading dose, and that dose times 7 and times another 2. So 100% of that dose initially is a loading dose in the loading dose arm. And the bottom line, what was seen, and we see here, is a greater percent of patients in range, so 70 to 180 milligrams per deciliter during the final 2 weeks of treatment, so week 15 and 60 on continuous glucose monitoring, when the loading dose was given. And when the loading dose was not given, a similar proportion of patients achieving that time in range with Icodec versus insulin glargine. And you can see here the fasting glucose throughout the 16 weeks. And basically what the loading dose does is it prevents this temporary rise in glucose when the loading dose is not given. And importantly, there was no increased risk of hypoglycemia with the loading dose. So this study basically confirmed that a loading dose is important to prevent temporary hyperglycemia, and there's no risk of increased hypoglycemia. And the second study looked at patients who were insulin naive, looking at different titration algorithms, either to a goal of 80 to 130, titrating by 21 or 28 units weekly if patients were not achieving the goal of less than 130, or a more aggressive algorithm, algorithm 3, to glucose of 70 to 108 milligrams per deciliter, as was done in the initial phase 2 trial, versus insulin glargine. And what was seen here is actually compared to insulin glargine, actually in each group, there was an increase from baseline to the end of treatment, so week 15 and 16, better with respect to time in range, and significantly better with algorithm B, so going to 80 to 130 with 28 units per week titration than insulin glargine, and similar in titration C and algorithm A, which was 80 to 130 plus or minus 21 units weekly, so a more conservative titration algorithm. And importantly, with titration algorithm B and C, so the more aggressive algorithms, greater level 1 and level 2 hypoglycemia compared to insulin glargine U100, particularly with titration algorithm C, which was the most aggressive. And with titration algorithm A, very similar with respect to hypoglycemia and glycemic control versus insulin glargine U100. And importantly, in all of these treatment algorithms, there was no level 3 or severe hypoglycemia. So what the authors concluded was that Icodec titration algorithm A, which was a target of 80 to 130 milligrams per deciliter, adjusting the insulin by 21 units per week as needed, displayed the best balance between glycemic control and risk of hypoglycemia. So to summarize the Icodec phase 2 trials, there was comparable glycemic control between the once weekly insulin and comparable safety profile compared to insulin glargine U100. An initial loading dose prevented transient rise in fasting glucose during the basal insulin transition without an increase in hypoglycemia. And a titration to a target of 80 to 130 milligrams per deciliter resulted in the best balance between glycemic control and the risk of hypoglycemia. And this is just a quick overview of the Onwards program, which is the phase 3 trials assessing icodec in patients who are insulin naive, insulin treated patients with type 2 diabetes, as well as Onward 6 in type 1 diabetes patients. So to summarize, the evolution of basal insulins over time has led to formulations with longer and flatter time action profiles, resulting in lower risk of hypoglycemia and less frequent dosing, so greater convenience for patients. There are 2 once weekly basal insulins that are currently in clinical development, basal insulin FC or BIF, which is in phase 2, and icodec in phase 3. And both have demonstrated comparable efficacy and safety compared with once daily basal insulin analogs. And there are clearly potential healthcare provider concerns about once weekly insulins, which include the risk of hypoglycemia, unfamiliarity with initiating and titrating once a week. So there'll need to be a lot of education, particularly given such large doses once weekly as opposed to spreading them out once daily, and the lack of dosing flexibility potentially. So once weekly basal insulin formulations have the potential to enhance adherence and persistence with insulin therapy and thereby may improve long-term patient outcomes. And it'll be very interesting to see results, obviously, of the phase 3 program and of Lilly's insulin completing phase 2 and hopefully going into phase 3 as well. That's sort of the new frontier in basal insulin therapies. Thank you very much.
Thank you very much, Dr. Frias. We now invite Dr. Kudva to speak.
Thank you very much. I'm grateful to the Endocrine Society for this invitation. This is an exciting year, and certainly we use this as inspiration to accelerate further discoveries and applications of discoveries. So I will be talking about game-changing insulin therapies for patients with diabetes. And it is good to start with looking at the unmet needs for people with diabetes. As we know, the 2 main types are type 1 and type 2, though other types are increasingly being recognized. So this is a slide showing data from the Type 1 Diabetes Exchange, which has provided a lot of information about patient-important outcomes and the state of treatment for type 1 diabetes in the US. So what you can see on the right side here in a slide is blue showing A1C trends for people with type 1 diabetes across the lifespan, more recently from 2016 to 18, and the orange showing the A1C trends about 6 years prior to that, from 2010 to 2012. As we can see, A1C still need to improve, and the trend is still not in the direction of as much improvement in the A1C as we would like it to see. So the challenge continues, and the reasons why may be different. So the reasons may be different in previous years versus currently. So on this slide, you can see 3 different age groups and 4 different depictions. So from left to right in each scheme, the cohorts represent the type of treatment and the use or the inability to use CGM, continuous glucose monitoring. So the right 2 bars are the use of continuous glucose monitoring, either with injections on the left side in each scheme, or with insulin pump on the right side with each scheme. So as continuous glucose monitoring is used, A1Cs improve. And with insulin pumps, there's probably more trend for improvement in A1Cs. So the summary here is that we need to keep working at improving glycemic control for our patients. And one way is to use more technologies such as continuous glucose monitoring. And another way is to use automated insulin delivery. And there are several components within each that could be improved. Both of these technologies will be addressed in further presentations to mark this occasion. So let's look at type 2 diabetes here. And this is a manuscript that was published earlier this year in the New England Journal. And on the left panel, one can see that A1Cs still tend to be suboptimal, even as we look at 3-year blocks. And this is the NHANES study, again, enrolling people in the US. And on the right panel, we can see various therapies that have been used in people with type 2 diabetes with the attempt to improve glucose control. Orange shows insulin use and the proportion of people on insulin. As we can see, that is now about a quarter or so of people with type 2 diabetes, but using it to improve outcomes is our mission. So it's good that various non-insulin therapies in type 2 diabetes have shown significant impact on target organ complications. Type 2 diabetes, as we know, is a very heterogeneous disease. So one of our challenges is to incorporate better insulin therapy within the framework of personalized medicine, something that all of us are vested in and need to expand. So the framework, therefore, for this talk is to look at both type 1 diabetes and type 2 diabetes and look at various attempts at novel insulin formulation development and testing. So within type 1 diabetes, these could be used in automated insulin delivery systems or with non-automated, such as injections or insulin pumps that are not automated. So the formulations are several, and they are listed here on the slide, and we will go over what we know at this time as also project towards the future one by one. So let's start with ultra-fast acting insulin. So in this slide, 2 of the preparations that are approved and available for use are listed, and also another one, the BioChaperone Lispro, which is in the process of being tested further. So the mechanism of action for these is similar and essentially involves quicker bioavailability from the site of injection broadly, though the various excipients that are present are different, and therefore at a micro level, the mechanism is different. So Fiasp from Novo Nordisk is the first such approved preparation. So on this slide is an attempt to show data for type 1 diabetes that was used and is now part of the FDA package insert. So we can see that mealtime Fiasp improved A1C more than post-meal Fiasp or mealtime Novolog. These were non-inferiority studies and ended up showing superiority for Fiasp. There has also been an attempt to use Fiasp in automated insulin delivery systems. So on this slide is a summary of 2 small studies which have done the testing. As we can see, these studies showed non-inferiority and for one, and for another showed some superiority regarding time in target. Again, one of the questions is which metric to look at. So if one looks at the metric of glucose control in the first hour after the mealtime insulin is injected, there is superiority for the faster acting. So the point to learn here is that this was done with algorithms that did not take into consideration pharmacokinetics and pharmacodynamics of ultra fast acting insulin. And so that is a point, that is a place for further development and testing. Lyumjev is the second approved preparation, also, and this one is from Eli Lilly. The FDA package insert shows data for both type 1 and type 2 diabetes. The scheme of testing is similar. The design is non-inferiority. And again, a mealtime Lyumjev is within this framework found to be superior to either mealtime Humalog or post-meal Lyumjev. The post-meal approach might serve people with decreased gastric emptying or gastroparesis. This also has been tested without algorithm modification in a 670G system, and at this time has been shown to be similar to Lispro insulin. There is, however, a larger effort here, the Medtronic 780G study with Lyumjev, which is going to be active with enrolling patients very soon. So we look forward to the results of this study. So this will have a more sophisticated algorithm, but still does not take the PK/PD of the insulin preparation into account. So what about hepatopreferential insulin? So one of the challenges with peripheral insulin delivery is that there is the inability to reach higher concentration within the portal circulation. So trying to address that have been two approaches: the basal insulin pegylated Lispro, the BIL preparation from Eli Lilly, and the hepatic-directed vesicle preparation. So the BIL development was stopped, as we know, a few years ago, and this was related to the fact that this had too much impact probably on the liver, resulting in some liver-related endpoints that were not considered suitable for further development. A different technology, as listed here, is used for the hepatic-directed vesicle. So this is a phase 2B study showing that depending on the A1C that is achieved, the incidence of severe hypoglycemia may be different. So when the A1C is higher than 8.5%, the HDV Lispro does better, but when the A1C is less than 8.5%, it does not do as good. There are several trials in type 1 and type 2 with this technology that have been completed. We await results. Developing oral insulin has been a quest now for probably 98 years. So starting soon after the development of or the testing of injectable insulin, various technologies shown here have been developed and tested. So the ORMD 0801 shown here in phase 2B tested different frequency of administration and different doses. And as we can see, 32 mg given once or given thrice came out as superior. This is a presentation from the ADA last year. And therefore, there is a phase 3 study now that is ongoing, enrolling people with type 2 and using this once a day. And we expect to have results in 2024. So we look forward to this. And this has certainly potential to be the first commercially available oral insulin. Tregopil is another oral insulin which has been tested. As we can see, there are PK/PD data for this. And so when it is given before mealtime, there are certain insulin profiles that are achieved. And there is some impact on rate of change of glucose. So obviously there is expectation about phase 2 and phase 3 studies with this preparation. These were non-inferiority studies. Results are awaited in the form of publications, but the results that are present on clinicaltrials.gov indicate that the impact so far has been modest. So what about oral basal insulin? So Novo Nordisk developed insulin 338, and this oral basal insulin was not shown to be equivalent to glargine when tested in people with type 2 diabetes with similar hypoglycemia rates, but the oral doses were high and production for widespread use was not considered commercially viable. So in this space, at least for this product, further development seems to be slow. So further technology development may be needed. So there is a self-orienting millimeter scale applicator technology, the SOMA technology, which is currently being tested, developed from MIT, and we await further news. Insulin co-formulations with pramlintide are an exciting area. We and others have done studies with pramlintide. We did them in people with type 1. Others have done it with type 1 and type 2. And as we know, for type 1 diabetes, this is the only non-insulin FDA approved preparation. What we can see listed here are products from Adocia and Xeris. And as we can see, various co-formulations are being tested at this time, and they are in phase 1-2. Some have been completed, but there is potential for this field to expand and get faster soon. So one such preparation is shown here, and the framework for testing is generally similar. Compare this to a rapid-acting insulin or maybe an ultra-rapid-acting insulin going forward, and also that plus pramlintide separately, and then test the co-formulation. The good news is that these co-formulations appear to be well tolerated, and there is potential for further testing. And the same is true also for Xeris. What you can see here is that these technologies are ready for further testing. Ericore is a third player and has developed both ultra-rapid acting insulins and also co-formulations. So these co-formulations have been tested in automated insulin delivery systems also. And the difference here would be that meal announcement is not done. So there is more automation here with a co-formulation. The initial studies show non-inferiority. So these could be tested further. Smart insulins have been another area of great endeavor. So what we see here is an attempt to show that a smart insulin, which has a linker that links to a lectin receptor molecule, and therefore here with high insulin concentrations, binding of the linker to the alternative receptor results in quicker offset and therefore less hypoglycemia. This is a great scheme, but when tested further, has not been found to be as promising. So further development here with alternative technologies is awaited. It's good to think that oral insulin could be used to prevent type 1 diabetes by causing immunomodulation. On the left side is a summary of the American studies. So again, oral insulin was not as active as we would like, not as effective as we would like it to be. But similar studies are being done in Europe with some design changes, a younger population being recruited, and an additional endpoint of impact on antibodies. And those will provide us with useful information as we try to continue this quest. So this is the timeline for type 1 diabetes. We looked at several developments that have happened and several developments that are going to happen in the next few years, and hopefully not just treatment of type 1 and type 2, but prevention of type 1. And similarly for type 2, there is testing along the lines of treatment, not along the lines of prevention. I use this slide to show that there has been a lot of quest to develop these preparations. Trial information is much less. So game changer progress has been difficult, but the endeavor has been fabulous. So we clearly do the study in partnership. So many of my colleagues all over the world have done these studies. Patients and families have participated. Funding agencies and industry have made these possible. Of course, here at Mayo, with our team, we try to play an active role. So I thank you for your attention.
Thank you so much, Dr. Silva. I'll turn it over to my colleague Jenny now to begin the Q&A.
Thank you, Colleen. Our first question is for Dr. Sequest. Dr. Sequest, can you talk a bit about what are the costs of the new glucagon drugs compared to the IM glucagon kits?
Well, the manufacturers have been very clever, at least in my area, where they are very much the same price. And so it's merely a matter of who's covering what is how patients really determine which product they get.
Great, thank you. Dr. Sequest, is glucagon generally accessible to patients in terms of affordability and insurance coverage?
Generally, yes. Some form of glucagon will be covered by most insurance plans. It may not be the one that they want to use, and we may have to prescribe something else. I think we can make a pretty good argument about some of the ease of use of the newer products and may be able to make some appeals to our insurance covered companies about that. As time goes on, I think patients will begin to use more of them, and we'll see how the patients view it. I imagine if they prefer one versus another, they will probably advocate for that with their insurance company.
Great, thank you so much. Dr. Frias, I have a question for you. Is it fair to compare hypoglycemic events with degludec to the BIF insulins when the targets were so different in the studies?
Yeah, so that's a great question. Probably not, because we did reach lower glucoses with insulin degludec, because the target was less than 100 as opposed to less than 120 and less than 140 milligrams per deciliter. So I think this will be teased out in the next phase 2 trials that are ongoing, where the targets are similar with the BIF group and the insulin degludec group. We did, though, do an analysis, and it was a post hoc and exploratory analysis looking at, irrespective of the fasting glucose that was reached, though, there was always less hypoglycemia with BIF. So this gives us an indication that that very, very steady sort of insulin pharmacokinetics may have an advantage. But no, we need to compare the two insulins with the same fasting glucose target to really get an idea whether there's an advantage in hypoglycemia with BIF versus degludec, and that is being done. So it's a great question.
Great, thank you. Dr. Kudva, can you tell us a little bit about, obviously, it sounds like the landscape for oral insulin is not as bright as people had hoped. What's the landscape looking ahead to the future? Are groups continuing to research it?
Yeah, so I think the one preparation, so are you able to hear me, Jenny?
Yes.
Okay, so the one preparation that I showed is the one most promising, so that was tested as once daily, twice and thrice daily, and I think two of the three ways of giving it are being tested further. So it might be launched as a once daily or a twice daily, but we're looking probably at 2024 before that is approved. The second preparation from India, the results so far from what I can see do not look promising enough for it to go to the next level, but there may be information others may have on the stock, so feel free to add that. I was just looking at every possible place I could get information from. So in opposition, counters to some of the other newer preparations being tested. For this, actually, a lot more results are available on clinicaltrials.gov. So it is public information to see. It's not been peer reviewed and published, but the information provided is sufficient enough to say that the effect is not promising enough to move further. So Biocon from India might be looking at improving it or modifying it. I think Some of these are some of these have been important lessons, I think, some of the preparations that have been tested. For example, the hepatic directed vesicles seem to be quite powerful working on the liver, but maybe too powerful. So there may be a bit of a scale back that needs to be developed.
Great. Dr. Frias, can you tell us a bit in the BIF phase 2 trial, why were the fasting glucose targets higher for BIF than for insulin degludec?
Yeah, so this relates sort of to the last question. And this was the first study, it was an early phase 2 study, and the first study in which BIF was being used in the outpatient setting. So it was really more of a safety concern. So we had the one algorithm where we were targeting less than 140 milligrams per deciliter, a second algorithm less than 120, and we talked about previously the degludec arm less than 100. And again, in the ongoing phase 2 trials, I should say now, the targets are similar and less than 100. So this was purely for safety reasons.
Okay, thank you. Dr. Frias, one more question for you. Is weekly insulin being tested in people with type 1 diabetes? And if not, why not?
Yeah, no, it is both BIF and icodec. So in the Onward program, which is Novo Nordisk phase 3 program, there is a study in type 1 diabetes, and Lilly's basal insulin FC is also being tested in patients with type 1 diabetes.
Great, thank you. I have a question from nutrition blogger Bea Paredes. She asks, is insulin resistance something that can be treated with just a low glycemic diet, or do you think medications are needed?
Well, I can start with that and see what my colleagues have to say. I don't think about treating insulin resistance per se. I'm, of course, a diabetes doctor, and I think about treating diabetes, and that treating diabetes with a well-balanced diet that is appropriate for the patient in front of me is how I think about it. I would not necessarily start a medication to treat insulin resistance.
I would agree, and I think we're focusing more and more on weight loss. I mean, the majority of patients with type 2 diabetes are overweight or obese, so certainly managing weight reduction, which improves insulin sensitivity, is very, very important. So it's a good balance. I mean, the cornerstone needs to be proper diet, physical activity, but a lot of these patients will need medication, as Dr. Sequist points out, to treat their glucose.
I agree. I have nothing to add. So thank you both.
Thank you. Dr. Kudva, what are kind of the next steps to seeing more progress with smart insulin?
Yeah, so I can use that to refer to glucose responsive insulins. So I think there is more development going on in this area. It's a fairly complex area. So Dr. Michael Weiss has written a very good review article that was published in Diabetologia earlier this year. And based on some modifications to what can be linked to insulin, they have a very good program that they are developing further. But that program is very early in its development. Now, there are 2 other products that are available to Lilly and Novo, and these were acquired by them from other companies. Unfortunately, we don't have details about the ways in which these smart insulins work. So we'll have to watch the space. The Juvenile Diabetes Research Foundation has been funding more work in this area. So there is more basic science going on, and especially from researchers at University of Utah in Salt Lake City. So I think there's a lot of development going on here, Jenny, but I think as the last slide that I showed, which is a review 7 years ago from Dr. Heinemann, I think that still holds good. I think the number of endeavors to try and develop more products is still 10 or more times the number of clinical trials that are done. So I think the development space here, and the development trajectory is still challenging. Getting something to a clinical trial is taking years, and then the results of the clinical trials have been modest at best, and sometimes have not resulted in further evaluation. For example, the Merck product, which was tested, it was tested in a very limited manner, and then the work didn't progress further. Though if you look at the preparations, more testing could have been done. The example I take is of insulin pramlintide co-formulations. You can see in the slides that there are several co-formulations that are being tested. I think that's a very good model to try and evaluate further products that are developed. It's also humbling to note that the insulin pramlintide co-formulations have taken about 20 years to come to this place. I think, Simlin was approved for type 1 diabetes probably close to 17, 18 years ago, I think, somewhere there, give or take a couple of years this way, that way. But we really should be thinking about, how can we accelerate this going forward? So I don't have any insights about game changer technologies which might be coming up in the next 2-3 years. But I think we're all hopeful that there's something out there being tested now or being developed that could do that. And when it does come up there to be tested, we look forward to doing it. I'm sure there are many others who are looking forward with the same eagerness.
Absolutely, thank you. Could one of you address the high costs of diabetes therapies, especially insulin? We know many patients need to ration their insulin due to high costs. How do we navigate that with these new technologies coming on the market?
Yeah, I think this is a difficult issue, right? I think at least in the US, it has been a very difficult issue for our patients. I think other countries do it better. I think there is a more recent manuscript in Mayo Clinic proceedings. It's either going to be released soon or it may be in online, about studying this in Canada versus studying this in the US. How has it been made available in Canada, and what are the challenges in the US? But the challenges in the US are clearly much more than in any other country. Perhaps others may want to add some points here.
Yeah, I think it's an issue with all new medications and with insulins as well that have been around for a long time. It's clearly a very complex issue. I think it's being worked on. There's certainly been a lot of attention placed, particularly on insulin over the last several years. And I've actually seen quite a bit of improvement. It's nice to see also the companies have patient assistance programs and those sorts of things. Most of my patients are grossly underinsured or uninsured, so we rely on a lot of different programs to give them insulin when they need it. So hopefully, with the attention that's being given by societies, by the press, by clinicians as well, things will get better. But the bottom line, these are expensive drugs to develop as well, and we need to keep developing them. So I think it's an important balance, and then hopefully it'll work its way through.
Yeah, I agree completely. I think this is one of the things that we can do as members of the Endocrine Society, which does have a whole advocacy group, as is true of other professional societies. We have to work side by side with our patients in order to change public policy so that we can come up with more affordable therapies for people.
Great, thank you so much. That's all the questions in our queue, so I'll turn it back over to Colleen.
Thank you so much, Jenny, and to all of our speakers, and for all those great questions. We're going to take another quick 5-minute break, and then we'll see everyone back again at 1:30 Eastern for our final session. Welcome back, everyone. Now for our final session on insulin technology and future delivery models. We have with us today Dr. Ann Peters of the University of Southern California, Dr. Stephen Russell of Mass General Hospital, and Dr. Derek O'Keefe of the National University of Ireland, Galway. Dr. Peters, we invite you to please speak first.
Excellent, and thank you for having me. So today I'm going to talk about advances in CGM. And first of all, I want you to keep in mind that I'm a clinician. So I'm showing you things that I thought were interesting clinically, but don't ask me for the technical details, because for that, I would ask an engineer. These are my potential disclosures of interest. And in reviewing this field, I think that the main themes in CGM development are the following. First, the devices by and large are becoming smaller so that their form on the body becomes easier and less obtrusive, and patients may like wearing them more. Some of them have become longer lasting, which is also a good feature. Others are more accurate. And then there's a number of different non-invasive options. None of these are ready for prime time, but they've always been something that's held a lot of interest for patients. When I was a fellow a very long time ago, I remember working on a non-invasive model, which obviously didn't come to fruition. But if they did, I think it would be really nice for a subset of our patients who really don't like the idea of poking anything under the skin. Increasingly, the devices are becoming interoperable so that pieces can be shared with other pieces, which I think is a great advancement. And then we're seeing the ability of these devices to measure more than glucose, which may be helpful and interesting as we move forward. The first device is the Abbott Freestyle Libre 3, and this is now available in Europe. I show it here, and you can see that it's the size of 2 pennies held together. So you can imagine that in your mind. And you can see on that human arm what it looks like compared to the Libre 2. So it's 70% smaller. You can see that it's even less thick. And from what I've read in Twitter and other kinds of social media accounts, people have been really liking it because of this smaller size. And it has been something that I think will be increasingly accepted. One of the most important features is that it doesn't have to be swiped. So it sends a continuous real-time glucose reading to a smartphone every minute. And then, much like the Dexcom, people can look at it, say, on an Apple Watch and see what their glucose levels are. There are optional high and low alarms. It has a MARD of 9.2%, and like the other Abbott Libre models, it lasts for 14 days. Next, we have a Dexcom G7, and this is not yet available, but hopefully will be soon. And going on to our coin comparison, it's about the size of a nickel, and it is somewhat thicker. And it is obviously much smaller than the Dexcom G6. But more importantly, I think it is an all-in-one device. So you don't have to stick on the sensor and then put on top of it the transmitter, but it's all in one and very easy to insert. And both the Libre 3 and the Dexcom G7 come with much smaller amounts of packaging. So both are much better for the environment. Instead of a 2-hour warm-up, it has a 30-minute warm-up. And it has a capacity to directly connect with other devices, such as the Apple Watch, or shown here with Garmin fitness devices. This is actually shown with the Dexcom G6, but it will also pair with the Dexcom G7. So people who want to can mesh all of these things together. It will also mesh with programs such as Livongo. So data will become easier to access, and for patients potentially on devices they may already wear and like, this will make it easier for people to see their information. In terms of Medtronic, we have the Guardian 4, and this is also available or will be available soon in Europe. And now it goes with the MiniMed 780G system, and it does not require finger sticks. And it's similar in form and function in many ways to the Guardian 3. So it's got what they call a seashell shape. It's hard to say quickly. And so it's not that different looking, but I think it is different in that it does not require finger sticks and is accurate and should be able to work well with the 780G system. Medtronic has also been developing other sensors, and these are called Zeus and Synergy. And Zeus will have an ICGM classification, which is good. It means it's at a higher standard than some of the other devices. And it does require calibration, but just once on the first day of use. And as I said, it looks like the other sensors, the Guardian 3 and 4, is that seashell design. Synergy, which is the next out in terms of development, is now going to be an all-in-one device. It's 50% smaller. And instead of looking like a seashell, as you can see in the illustration, it's a flatter, thinner device, and it also requires calibration just on day one. So this now becomes a different looking, feeling device than the prior series of Guardian sensors. Eversense is developing, or has actually developed, a sensor that lasts longer. So it's the Eversense XL. It's a 180-day sensor. It's available in other countries and will hopefully be available in the United States in the near future. And it extends the ability of patients to wear this so that there doesn't have to be an every 3-month reinsertion. Now, moving on to more sort of fantasy approaches, as I said, these are non-invasive sensors. So the Apple Watch has long been touting the fact that it wants to be able to measure all sorts of things, including glucose. And there are rumors that in the next generation Apple Watch Series 8, that there will be an ability to measure glucose. I couldn't really find specific details about this as to how this would happen, but somehow it's supposed to measure glucose. This is interesting, and we'll see what happens, but at least we will have other devices where data can be shared with an Apple Watch. We already have the Dexcom that can do that. And once the Dexcom G7 comes out and there's a direct feed to the Apple Watch, I think it will make it easier. And who knows what Apple will be coming up with. But devices under the development process include these. So there's a company called No Labs, which is kind of an interesting name. And they have a proprietary technology, which they call BioRFID, and that stands for Body Radio Frequency Identification. And this is a non-invasive diagnostic technology platform, and it uses electromagnetic energy in the form of radio waves to non-invasively capture molecular signatures, including glucose. And what's really cool about a device like this is there's no consumables. So you basically have the device and you use it, and I guess it would wear out at some point, but you don't have to keep opening a box and sticking something on and throwing it away. And this is what they imagine their device would look like. And it's something that you could either hold in the palm of your hand or put on your arm, or however you wanted to do it, that would be able to give you the glucose levels. And they have published data. And here is the data that they have comparing it to the Accu-Check. They've compared it to the Dexcom. They've compared it to the Freestyle Libre, and the curves look pretty good. So in the blue, you can see the system measuring glucose, and that's compared to the AccuCheck. So it's nice. It looks to me like it has potential, but they haven't yet made it look like that first slide I showed you. So when they did this study, it looked like this. And basically that red box down there that's taped down with duct tape is the thing, that's the device. And people had to rest their elbow on the arm of a chair and then put their arm down on it, and then it made the signal that you saw in that graph. So it did start out much bigger. I think it was 3 feet by 3 feet. So they've gone a long ways in terms of miniaturizing it, but they still need to make it a lot smaller so that it can actually be used by humans. So I think that the technology works, but we have to have it in a form that makes some sense for people to wear. Then there's a whole series of non-enzymatic glucose sensors that make a certain amount of sense compared to glucose oxidase sensor, which is what we're used to. Theoretically, these could outperform the enzymatic ones that we're used to because they'd be cheaper, they'd have enhanced sensitivity, they'd be more stable, and they would last longer. But again, these are only under development. And there's a form of a laser-induced graphene or LIG sensor. And these are a form of laser-induced nanomaterials with high electrical conductivity. And you can see in the graphic on this slide how they basically make these. But it turns out that it doesn't actually measure glucose directly. You have to combine it with nickel and gold. And remember, lots of people have nickel allergy, which is part of why they plate it with gold. And then this measures glucose in sweat, not in interstitial fluid. And they've published some data that actually looks reasonably good at how it measures glucose in sweat. And you can see that little prototype of the device. So it is already small, but it has to have a lot of testing to prove that this would work, again, at measuring glucose in human beings. There's another company that I found, which is called Graphware. And these are their mock-up illustrations of what this will look like. And it's another graphene-based skin surface wearable. And it's actually a little film that you put on your skin and then the device on top of it. And they want to be able to measure multiple disease states, including glucose levels. So we'll see what this comes to end up working like if it does come to market. Something much closer to coming to market is the Abbott Libre ketone monitor. And this now isn't measuring glucose, it's measuring ketones. And what this does is to provide episodic beta-hydroxybutyrate values. And I personally really want this to come to market. And I believe it's going to be just like the glucose Abbott Libre sensors, in that it's going to be a round disc that then you swipe and you can see what the ketone level is. And this is data that they obtained from a study. And they did this study up in Santa Barbara with 12 volunteers who didn't have diabetes, but were on a ketogenic diet. And this is just tracing from one subject who was wearing 3 sensors. And the red triangles are the reference measurements of the beta-hydroxybutyrate. And you can see that it is pretty darn accurate. It goes up and down pretty closely to what the reference points are. And I really want this because I really like using SGLT2 inhibitors in people with type 1 diabetes, particularly for their cardiac and renal benefits. But I don't even know what day in, day out ketone levels look like in any of my patients. And so I would have to learn a lot clinically about what a device like this looks like, but I think this has great potential. And I'm very glad that Abbott is developing it. Then there are other things that are being measured. So this is a continuous lactate monitor. Again, it's a non-invasive monitor. It's under development in Sweden, and their target market are athletes. I don't know if this is going to be really useful, but athletes, at least certain high-end ones, are very interested in lactate production, obviously because it affects performance. So this has some potential for those individuals, and it may be interesting. to see what the data shows. And since I've taken care of at least one and now two Olympians, I think at that level of sport, it will be helpful. And then there is this, which is a stretchy polymer. And I think it kind of looks like a form of modern art, but this is being developed at the UCSD Center for Wearable Sensors. And it's the size of a postage stamp, and it measures everything. So in the middle, you can see that horizontal row of sensors. It measures blood pressure. And then one side of it measures sweat, and one side of it measures interstitial fluid. And the side that measures sweat measures lactate, caffeine, and alcohol. And the part that measures interstitial fluid measures glucose. Now, I personally don't want anybody to tell me how much caffeine I have in my blood because I need just enough to function. But there are other things that are going to be much more useful, and in particular, things like alcohol, so that people can see what their levels are before they do things like have another drink or go out for a drive. So I think this, again, is something that's fascinating to me. I think it has a great deal of potential, and let's see if it comes to market. So in conclusion, the existing CGM devices are always in a state of evolution. I always want the next better version. So do my patients, but I'm really thrilled that I think in the next year or two, we'll really see advances in the development of CGM for our patients. I think that accuracy, ease of wearability, and the ability to interconnect are very important features. There are non-invasive sensors being developed, which again, patients really love the thought of, but I'm still not convinced that those are close to being available for patients. And then I think that these devices are going to start measuring more things than glucose. Some I think will be more useful than others, but I'm very much looking forward to the ability to measure ketones in my patients, particularly with type 1 diabetes. So with that, I thank you very much for listening. Thank you.
Thank you so much, Dr. Peters. We now invite Dr. Russell to please speak.
Thank you very much. I'm pleased to be here at this meeting that is honoring the 100th anniversary of discovery and availability of insulin. And I've been asked to talk about the future of insulin delivery. These disclosure slides often go by pretty quickly, but I'm going to spend just a moment on this because I think it's important to note that I have ties to a company that I'm going to be talking about during the talk. I've been working for quite a number of years on one of these automated insulin devices called the bionic pancreas, and have a number of patents that are assigned to my employer, MGH, and licensed to the company. And I also consulted for the company and have had research support from the company in the past. So of course, what we're talking about today is automated insulin delivery. This is something that has been thought about as a goal for a long time and recently has become a reality and has become available to patients. But the idea is to take advantage of all of this glucose information we have from the new continuous glucose monitors. And instead of relying on the person with diabetes to make decisions based on that information, instead have dosing algorithms that use that information to dose insulin, pumps that talk to those algorithms and automatically deliver doses. Of course, there's still going to be patient-specific factors that affect how that insulin works. But the fact that you get 288 chances every day to adjust the insulin dosing up to every 5 minutes means that there's the potential to dramatically improve glycemic control with these approaches. And to use all of that glucose information coming from the CGM that no person could really attend to throughout 24 hours a day. There's quite a number of companies that are working in this space, and several of them have actually brought products to market, and others are planning to bring products to market soon. I can say that in some sense, the future is already here because there are a number of these devices that have already come to market. This is one that's available in the United States, the tandem t:slim pump with Control-IQ. This company did a randomized control trial that was actually not done by the company. It was done by investigators and funded by the NIH. And this is to date the only randomized control trial that's been done that led to approval in the US. And they were able to show that the use of the closed loop feature of the device significantly increased time and range. It decreased mean glucose and led to statistically significant reductions in time less than 70 milligrams per deciliter and time less than 54 milligrams per deciliter, although in particular, this was a modest improvement. And this study was done in 168 individuals over 6 months and looked at 14 to 71 years of age. And there's been subsequent studies that looked at a younger age group with this device as well. There's also a device called the 670G from Medtronic that's been approved for some time, but Medtronic now has a new device or a new generation of this device called the 780G that has undergone a pivotal trial and is currently being considered by the FDA. This device has a number of improvements over the 670G, which is currently available. One of them is that it allows more flexibility in the glucose target that's used. One of the concerns about the earlier device that it had a tendency to exit from auto mode fairly frequently and required quite a few finger sticks, and this device requires fewer finger sticks and has been shown to have fewer auto mode exits. Unlike the tandem device, this was not studied in a randomized control study. It was rather a single arm study where the 3-month intervention period was compared to a run-in period. So they can't exclude study effect, but at least compared to that run-in period, there was an increased time and range, decrease in mean glucose, and no statistically significant differences in the amount of hypoglycemia, but certainly fewer auto mode exits. Another device that has been studied and is currently under consideration by the FDA for approval is what's called the Insulin Omnipod 5. This is unique among the other devices in that it uses a pod rather than a tubed pump. And this device also allows some selection of user-defined targets over a fairly broad range, although in the trial, most of them used the lowest target available. Again, this was a single-arm pivotal trial in 235 participants over 3 months, and they had 2 separate cohorts in adults and children. And similar to the previous studies, in the closed loop period, there was a lower mean glucose than in the run-in period, increase in time and range, and in this case, no differences in the amount of hypoglycemia. So it's expected that both the new Medtronic device and this Insulet device may be approved sometime in the near future, and that will mean that there are then 3 automated insulin delivery devices available to patients, one of which uses a pod and 2 of which use tubed pumps. Finally, this device has been pending for a long time before the FDA, but this is a do-it-yourself or user-developed system called Loop, and the algorithm runs on an iPhone or potentially any phone, really, a smartphone, and then it communicates to a pump using a communication technology that depends on the pump. And this has not been formally studied, wasn't developed in the academic setting, but rather developed by a number of dedicated users, but has now been studied in an observational study. And in this one subarm of the study, they looked at new users who were enrolled within 7 days of enrollment in an online format, and they compared their control historically to their control for 4 to 6 months on the device. And again, there was an increase in time and range, a slight reduction in mean glucose, and no changes in hypoglycemia. It's worth noting that quite a few of these studies have shown benefits in people who already had quite good glucose control. It's unclear how they would work in people with worse glucose control at baseline. There's a number of factors that determine the effectiveness of these systems, one of which is the appropriate choice of the system for the user, and as more systems become available, there'll be more options. Another is that all of these systems require at least some information about the insulin dosing parameters, either some or all of a basal rate profile, a carbohydrate ratio, sensitivity factor, or a total daily dose. Not all systems require all of these elements. But the elements that they do require, it's really important that they be fairly well optimized before the closed loop system is started. And then these insulin dosing parameters may need to be adjusted at intervals. And there's also user-related factors such as their diet and exercise. People who eat fewer carbohydrates tend to do better. How often they announce they bolus for meals. So all these devices that I mentioned are hybrid closed loop systems. So the user counts carbohydrates and determines the amount of insulin delivered for the meal. And then the system will manage basal rates, and in some cases will also deliver automated correction boluses. But the number of correction boluses delivered by the user can have a significant impact on the glycemic control. And what's important when thinking about that is that the accuracy of the carbohydrate counting by the user also matters quite a bit. And there's a lot of data out there that suggests that people aren't actually very good at accurately counting carbohydrates. And so if they overestimate carbohydrates, that can lead to hypoglycemia. If they underestimate them, that can lead to hyperglycemia. So to try and deal with some of these limitations, collaborators of mine, Ed Damiano and Faraz Al-Khatib, who were working at Boston University at the time that we started collaborating, had a vision for a new kind of automated insulin delivery system that would be different. And it had a number of design parameters that they wanted to incorporate. One is that it wouldn't require any information about the patient's insulin regimen prior to use of the device. So it uses only the patient's weight for initialization. That gives an initial dose scaling, and then it adapts online to their insulin needs, or that is the intent. And the user experience was intended to also be different in that the meal announcements would not require carbohydrate counting, that they could be announced just as the type of meal, for instance, breakfast, lunch, or dinner, and the rough size of the amount of carbohydrates in the meal, not carbohydrate counting, just as this the usual amount of carbohydrates for me, less than usual or more than usual. And then the system adapts over time to the amount of insulin that is needed for meals announced in that way. In addition, all the insulin corrections would be fully automated. And in one configuration of the device, it would also be capable of delivering small doses of glucagon for automated hypoglycemia prevention and treatment. The reason for that last is that when the glucose is well controlled, let's say that that the CGM glucose is right in the target range of 100 milligrams per deciliter, and someone starts to exercise, the blood glucose can fall fairly quickly. And all of these automated insulin delivery systems in a situation like this would immediately stop delivering additional insulin. The problem is that insulin is absorbed relatively slowly and then cleared relatively slowly. So the levels of insulin in the blood fall fairly slowly after the suspension of insulin delivery. And so but the glucose falls relatively quickly, for instance, in the setting of exercise, or even in the setting of an additional insulin bolus that was above the needed amount. And so the hypoglycemia will occur before the insulin levels fall very much at all. Well, how does the normal physiology deal with this? Well, the pancreas, when it's normally functioning, doesn't rely only on insulin. It relies on an additional hormone, glucagon. And when the insulin levels drop after the pancreas stops making insulin in the setting of hypoglycemia or incipient hypoglycemia, this de-represses the production of glucagon. And that glucagon can then go act on the liver to antagonize the effects of insulin and even turn the liver into a net glucose producing organ by causing the breakdown of glycogen and the release of glucose into the blood. And in people with type 1 diabetes, these alpha cells become dysfunctional and they no longer respond to hypoglycemia by producing glucagon. So the idea is to use both the insulin and glucagon as control arms to control blood glucose. And the idea here is that in this setting where the CGM glucose starts to fall, a system like this would suspend insulin delivery. That wouldn't be enough to prevent hypoglycemia potentially, but then you could give small doses, microdoses of glucagon, and that glucagon will then be absorbed much more rapidly than insulin. It has a time to peak in blood on the order of 20 minutes as opposed to 60 to 90 minutes for insulin. And it also acts relatively quickly. It can start to act within 5 minutes of delivery. And so that can counteract the effects of insulin and prevent hypoglycemia. If more is needed, more can be given. And one could potentially avoid the need to take carbohydrates to prevent and treat hypoglycemia. There are a number of companies that are working on bihormonal systems that use both insulin and glucagon, companies and academic groups. And this particular one is the one that I've been involved with over the years. Originally a collaboration between my collaborators, Ed and Faraz at Boston University and our group at Mass General Hospital. And now Ed and Faraz have spun out a company, Beta Bionics, to commercialize the system. This is a comparison, one of the studies that we did, comparing usual care in pump users, and most of them also use CGM, to the insulin-only configuration of this device and a bihormonal device. I should mention that this was an earlier version, not a version that's currently being developed for commercialization, but an earlier system developed by the Boston University group. And what we found in this study was that the insulin-only version of the bionic pancreas could lower mean glucose and increase time in range, but not alter the amount of hypoglycemia. So this is very similar to the other insulin-only systems that I talked about that are either already approved or are currently being considered for approval. The difference, of course, being that this system doesn't require any carbohydrate counting. In this study, this relatively small feasibility study, I should mention, there was no carbohydrate counting and all the insulin delivery was automated. So there was all automated correction goals. It wasn't possible for the individuals to give themselves additional insulin. Then when the bihormonal configuration was used in this study, there was a further reduction in mean glucose, a further increase in time and range, and interestingly, an even further reduction in the amount of hypoglycemia that occurred because of the addition of the glucagon. I should also mention that that study used glucagon, but it had to be reconstituted freshly every day because glucagon is quite unstable once it's reconstituted. And it was necessary to have a stable version of glucagon. And a company called Zealand Pharma in Europe has developed an analog of glucagon, similar to the analogs of insulin. In this case, the analog was designed to be more stable in solution. And so it's much more stable than glucagon. It has activity and side effects comparable to glucagon. It's now FDA cleared for hypoglycemia rescue and an auto-injector and is now being studied in the bihormonal bionic pancreas as a counter-regulatory arm to insulin. The company, Beta Bionics, has now developed a device that is capable of delivering either insulin alone or both insulin and glucagon, called the iLet. This is, I should mention, an investigational device, and it is not commercially available. It is the XY dimensions of roughly a credit card and is about 1.5 centimeters in thickness. It uses a Dexcom CGM, and it uses a cartridge for insulin and a pre-filled cartridge for this glucagon analog called dasiglucagon. And these are not compatible with each other. So it's impossible to mix up the insulin and glucagon cartridge. And then it uses two infusion sets in the bihormonal configuration and one in the insulin-only configuration. I am currently directing a pivotal trial of the insulin-only configuration of this device. And this is a roughly 400 participant trial, age 6 and up, a 3-month randomized control study, again, in this case, sponsored by the NIH. And it has a control arm that is usual insulin delivery plus the Dexcom CGM if the participants were not already using it. And the last participant in this trial was randomized in July. Coming up, we are also going to be running a pivotal trial of the bihormonal version of the bionic pancreas that will include roughly 700 participants, again, 6 months and up, or age 6 and up, and this will be a 6-month randomized control study with an additional 6 months of exposure and an extension study. And the primary outcomes of both of these studies are HbA1c, with the key secondary outcome being time in hypoglycemia. So looking further forward, what can we do further to improve these automated insulin delivery systems? Well, one of the key problems that all of these systems deal with is failed infusion sets. So when the device can't deliver insulin, there is the risk of hyperglycemia and ketosis. So we really need more reliable infusion sets, and a number of companies are working hard on developing these that would be less likely to fail in delivering insulin, and also could potentially be worn longer than the 3 days that all of these infusion sets are currently approved for. Another big improvement would be interoperability of components. So far, all of these systems use a single CGM, but not everybody can use every CGM for a variety of reasons, perhaps because they have irritation with the adhesive that's used. They can't keep them on. And there are a number of different CGMs available, and it would be great if individuals could use their CGM as long as it was accurate enough to drive closed loop with all of these systems. It would also be useful, for instance, to have different algorithms on more than one platform. So if someone did really well with the Control IQ algorithm but wanted to use it on a patch pump, it would be nice if that were possible. It would also be tremendously helpful to have faster insulin absorption and clearance. As I mentioned, the slow absorption and clearance of insulin is one of the limiting factors in how tightly glucose can be controlled and how much hypoglycemia can be reduced. It appears from some early data that adjustments to the algorithms may be necessary to realize the maximum improvements with better insulin absorption. It would also be helpful, even though it's already fairly rapidly absorbed, to have glucagons that are absorbed even more rapidly. And it may be useful to have additional inputs. For instance, continuous monitoring of ketones would be very helpful because it might tell us early on whether an infusion set had failed. One of the key markers of a failed infusion set is the increase in the ketones. And that would be tremendously helpful for these systems, and possibly even continuous monitoring of insulin levels. And finally, it would be really helpful to have further simplification of these systems for the users. Not having to count carbohydrates is great, but not even having to announce meals would be better. And full automation is, of course, the ultimate goal for these systems. That probably requires much faster insulin absorption and may require a bihormonal system with glucagon to get into a truly automated mode. So I think we've already made a lot of progress by having automated insulin delivery systems. I've seen how helpful they can be for patients in the clinic. Many patients have described the use of these systems as game changers. And we hope that there'll be further improvements that will really make it much easier to deal with the relentless nature of living with diabetes. Thank you very much.
Thank you so much, Dr. Russell. We now invite Dr. O'Keefe to speak.
Hello, my name is Professor Derek O'Keefe. I'm speaking to you today about robots, drones, and AI in diabetes care. We're looking forward to see what the future is going to bring us in terms of technology. I work as a clinician at University Hospital Galway, seeing patients every day, but also professor of medical device technology. So I'm really interested in medical devices. Here's my corporate research funding that I'd like to disclose. And then I guess you're wondering why I was asked to speak to you today about the future of this technology. I trained originally as an engineer, and engineers are quite logical. They're quite problem solving. Everything's black and white. And then, as I mentioned, I'm a clinician as well. So I trained as a physician. And everyone who practices medicine knows nothing's ever black and white. It's always a spectrum of color, and there's lots of clinical problems. So this mix of engineering and medicine, it allows great cross-pollination of two different fields to what I call a physicianeer, somebody who's able to use technology to improve patient outcomes. So I want you to think about for a second, what do you think is the most powerful tool in medicine? So if you think back through the history of medicine, what have been the great advances? Has it been hygiene, for example, when we figured out if you wash your hands, people live longer after an operation? Is it antibiotics when Fleming discovered penicillin? You know, tens of millions of people have survived longer because of that. Vaccines, we've seen how powerful they've been in the last year with COVID-19. But, you know, before that, they'd saved tens of millions of humans as well. You could argue that therapies such as insulin that we're celebrating today have saved and improved countless lives since their discovery. But my thesis is that going forward, the big tool in medicine, the most important one, is going to be data. And that's going to transform the way we practice medicine and the way our patients' lives are improved going forward, especially in diabetes, which is a very number-centric condition. I want to share this animation with you. Often what we consider valuable on Earth is given a monetary value. And this is the top 10 companies in the world over the last decade. And about a decade ago, what we considered most valuable on planet Earth per market capitalization was the petroleum companies, the hedge fund companies, the banks. These were the biggest companies in the last decade. But what's happened is in a very short period of time, it's flipped. Now the most valuable companies on planet Earth, as you're going to see, are the data companies. And you have to remember that Facebook was only discovered in 2004, YouTube was only developed in 2005, Twitter 2006, and the smartphone was only released by Steve Jobs in June of 2007. So in a really short period of our history, what we consider valuable has completely turned upside down. And the question is why? And the answer is very easy, is that data is valuable. In particular, what you've done before you've come to this present moment. And the best example of that is the internet. So when you come to a bookstore website on the internet, they don't really know what ad to give you, but if they know the last 4 websites you've been on, the so-called data exhaust or the cookies that you've expelled, then they can see, you know, that maybe you're looking at a medical textbook, you're looking at reading about burnout, then maybe you're looking at a website about art galleries. And so when you get to the bookstore, it says, would you like to buy a book on art galleries in Paris? Because it thinks you're going to take a holiday. So that's what made data so important in the last 15 years from a monetary point of view. And that was true for commerce and for banking. And the next decade and beyond, the great mine of data will be healthcare data because of the technologies we're going to talk about now and because of some of the stuff you've seen by my peers today. Just taking it back a step, why are we able to do this now in 2021 and, you know, not 20 years ago or indeed 100 years ago when Banting and Best discovered insulin? And the reason is all about this curve. This curve shows the computing power of what we're able to do in the last 100 years or so. And you'll see that it's increasing exponentially, the amount of arithmetic that we can do, but there's an inflection point on the graph right in the middle here in 1943 or 4, right in the middle of the Second World War. And unfortunately, with humans, we tend to innovate rapidly when faced with, you know, big challenges such as COVID-19 last year, but in this case, in World War II. And what happened was there was a young mathematician called Alan Turing from Cambridge who was tasked to create a machine that could crack the Enigma cipher, which was the cipher that the Nazis were using to spread messages and to get U-boats to sink Allied ships. So he created the world's first programmable computer in the 1940s. And from that then revolutionized humans in the fact that we could do so much more arithmetic with these programmable computers. There's a really good movie called The Imitation Game with Benedict Cumberbatch that tells that story. It's worth a watch. But right after he discovered the programmable computer and laid out the architecture of it, in the 60s, we developed the transistor. And then beyond that, in the 70s, we developed the integrated circuit, which allowed us to put millions and now billions of transistors on 1-inch piece of silicon that, you know, pretty much has stayed the same size. It's the density is what we've been able to increase. And, you know, most visually, we've seen that in our everyday life. If you think of the mobile phones that first came out in the 1980s, they were about the size of a brick. And they've got smaller and smaller and smaller over the years because of the density increase in the electronics. In a very short period of time, 15 years, the smartphone has replaced all these different discrete devices. And in fact, a cell phone could be as small as your fingernail. There's actually pendants out there you can get that do have cellular signals for SOS situations. But the reason the phones are still pretty big in our pockets is because when the smartphone came out in 2007, it was so powerful, you could actually connect to the internet, to early Wi-Fi, 2G, 3G signals. And what they realized then from a consumer point of view, rather than humans wanting the phones to get smaller, we wanted bigger screens so that we could watch cat videos, and therefore the phones got bigger. But from a technological point of view, they're remarkable, powerful computers that sit in your pocket. And because they're plugged into the IoT or Internet of Things, it creates a remarkable ecosystem that we live in. In medicine or in healthcare, we call this the digital health era. And this is this concept of this vast ecosystem of intelligent devices connected together over currently 4G networks and soon to be 5G networks, which are going to be even more powerful, both speed-wise, latency-wise, and connectivity-wise. And what that essentially means is that we're living in a connected world whereby your smartphone can get values from your glucose sensor and upload it onto a cloud, where, for example, an algorithm can do complex arithmetic to figure out and predict future glucose trends. So this is the world we're living in now, the digital health era of the 2020s. And in particular, when we think of diabetes, there is this term digital diabetes. And some of my peers today have talked about the amazing technology that's available to patients to look after their chronic condition, to monitor their glucose, and moving away from the discrete 4 times a day glucose to more continuous glucose monitoring, to insulin pumps, to devices that are connected, such as artificial pancreases, where the sensor can talk to the pump through an algorithm. It's been a remarkable time for patients with diabetes in the last 5 or 10 years, and it's only getting started going forward. The first thing I want to talk to you about is diabetes robots. And when you think about a robot, this is probably the picture in most people's heads. And when you think, what could a robot offer somebody with diabetes? And these are the kind of concepts. They could be a disease manager, they could be an educator, they could be an advocator, they could be a communicator, and they can be an emotional support to the person with diabetes. And to let you know, this research has already started because of the power of microcomputers and the availability, because the more that the computer shrinks the technology and becomes more powerful, and then they figure out the manufacturing side of it, the cheaper the actual technology gets. And we know that every year that, last year's mobile phone is cheaper than this year's mobile phone. So with this technology over the last decade, you've seen here in the literature and in some clinical practices, the use of desktop robots to interact in particular with children, to engage them in their care, for example, in type 1 diabetes. There's been some really good work done in this space. And now what we're looking at going into the next decade is moving that technology to a wider audience, adults with type 1 diabetes and type 2 diabetes. So this is an example of a robot that we've developed at my own lab in Galway, the Hive Lab, Health Innovation via Engineering. And it's based on a hardware architecture called Pepper Robot. And with this hardware, then it's essentially a blank canvas. You can program this robot to do anything that you would like. And that's what we've been working on with my team here in Galway. And we've done clinical trials with this robot that we've called Dave, the Diabetes Audiovisual Educator. And I'd like to just share with you a video of Dave so you can get a feeling of the interaction and how positive patients have been getting their diabetes education from Dave.
Who are you?
My name is Pepper. I am a soft-bent humanoid robot developed by Professor Derek O'Keefe and his Hive Lab team at the National University of Ireland, Galway for healthcare education. I was born in 2021 with a mission to improve human health. My first job is to remind you to clean your hands regularly so we can prevent diseases such as COVID-19. I can also demonstrate to you the correct hand washing technique. For my next job, I am currently learning about diabetes so that I can advise people about healthy eating choices and lifestyle changes to help them live longer. I believe that digital health is the future of medicine. Now please clean your hands. Nice to meet you. Enjoy the rest of your day.
So we used Dave in our clinical environment in the hospital this year, and we showed initially when he was deployed for hand hygiene that he increased hand hygiene by 20% at the entrances of the hospital and the clinics. And then we reprogrammed Dave, and we used Dave in the clinic, in the diabetes clinic, to engage with patients about healthcare education. And again, we saw a positive benefit of Dave teaching people about hypoglycemia management, teaching people about the correct diet to use. And we saw that Dave was as good as a clinician, a nurse, a doctor at imparting that information to patients. And this is a fascinating thing. You know, there's a whole area of academia called pedagogy, the study of teaching and learning. And we know how best to teach, for example, a primary school child or a 15-year-old child, or even, you know, a university college student. We know as humans how to teach other species, like, you know, how to teach a dog a trick or how to teach a dolphin to do something. But this area of using robots to teach humans, that's a whole new area of research, and it's really exciting, the area of patient education. So we're going to see a lot of this going forward into the future. And what it'll mean, in a sense, it won't be for everybody, but obviously with everyone living longer due to the success of medicine, we have this tsunami of chronic disease, and we don't necessarily have the clinical workforce to meet it. So, you know, when the patient is agreeable, they can get their education, for example, from a robot like Dave, and that frees clinicians up to do, you know, more higher value work on their license, like, you know, medication management and so on. This is another project I wanted to share with you. And again, it came from a real practical situation where I practice medicine in Ireland, we had a really bad winter storm whereby a lot of my patients couldn't come to my clinic. And as well as that, they were snowbound, which meant, you know, with patients with type 1 diabetes, they need insulin, and they didn't have a way to get to their pharmacy to get their insulin. So it made me think about ways of delivering insulin in the time of emergencies, such as Hurricane Katrina, or indeed in cases such as a pandemic now when communities are isolated. So this led to what we call the diabetes drone mission, which I'd like to share with you now.
I came to the island 31 years ago. I'm insulin dependent.
We make medicine for people with diabetes.
Access to this medicine, it's more than important, because without it I would be dead.
It's going to be a great mission.
We had a really productive meeting there with all the project team for the diabetes drone, the Sky Tango, allowing us to coordinate the actual mission between all the stakeholders.
Novo Nordisk, whose medicine will be part of the payload today.
A Wingcopter, who are giving us the actual drone that's going to fly to the island.
Vodafone, who are going to be providing the drone data link connectivity.
And Survey Drones Ireland, who are actually going to execute the flight for us for this innovative beyond visual line of sight flight.
The mission is just taking off.
The diabetes drone project was centered around delivering medications to the Aran Islands.
It's absolutely unreal.
Visual contact. Really amazing to do this, the first in the world delivery by BBLOS of diabetes medicine.
Everything is here that we need.
Fantastic.
So that was an example in 2019 of delivering a medication, BVLOS, beyond visual line of sight. And up to that point, internationally, the only people that are really doing beyond visual line of sight or beyond horizon drone flights are the big military organizations of the world. It certainly wasn't done in civilian airspace. So what we were able to show was, you know, getting over the regulations of airspace, getting over the regulations of medication prescription and delivery and the cold chain. It was entirely possible. And not only could you deliver a medicine, you could take a blood sample from a remote island community back to a major tertiary referral hospital. And what we're going to see in the next decade is going to be the explosion, the Cambrian explosion of drone technology to deliver a lot more things in our daily life, but in particular medications, because medications, as you know, are low weight and high value. And in the case of insulin, because it's a critical medication for people's life, it's going to be something that we're going to need alternative ways of having it delivered, especially in emergencies, but it'll become more routine over the next decade. The next thing I wanted to speak to you about is artificial intelligence. So we've heard from some of our colleagues here today about the continuous glucose monitor technology, whereby, you know, traditionally 4 blood sugars a day, now you're getting over 300 blood sugars a day. And when you don't see patients for a couple of weeks or months, that's again like drinking from a fire hose with the data when you're presented the data as a clinician. So the technology now is starting to evolve of the AGP, the ambulatory glucose profile, where this data is put into a harmonized format. And that's just to report it to the clinician. But the real interesting stuff is going on underneath the hood, the artificial intelligence that's looking at that data and then making predictions about the patient's future glucose trends. I've put up here an example of some of the papers that have been published in the last 30 years with artificial intelligence and diabetes. And you can see there's been an explosion, especially again in the last 10, 15 years, as that computing power and that connected world that we're living in now has come to fruition. And major companies have taken big steps in this area to try and predict your future glucose, 4-6 hours ahead of where you are now. And that's really remarkable. And they're able to do that because essentially they're looking at pattern recognition. And in some ways, certainly in the engineering world, when we hear the word artificial intelligence, it's kind of a buzzword for anything to do with computer patterns. But what it really should be called is machine learning, because that's essentially what we're doing with the technology. We're looking at patterns in the data, and then we're using machine learning tools to extract features in that data pattern. The uses are immense across diabetes, as they are across medicine, from the diagnosis to the management. So for example, you can apply a machine learning tool, and it has been done, to population data of weight and height and ages and so on. And you can stratify people who may be at risk for diabetes. And then, for example, you can target biochemistry testing on them. You can use it for clinical decision support, such as for insulin calculation apps, to predict hypoglycemia, as we mentioned. There's a big push now to have more equality in healthcare. So whether you're getting your healthcare at a major medical university hospital or you're getting it in a remote part of Africa, if you have an AI tool that can take a picture of the back of the eye, and then from there you just use a standard piece of code that can give you the same results as somebody in a major university hospital, that's going to have tremendous benefit for patients around the world. And then self-management tools as well. There's been a lot of talk today about the artificial pancreas system and so on, whereby algorithms are used for combining the glucose data with the sugar data with, for example, movement or food data to try and get better blood sugar values. So I'll leave it there. I want to thank you for your attention. The future looks bright for technology in type 1 diabetes. It's an area that's going to benefit first from this technology because of the advocacy of patients and because of the interest of the clinicians in this area, because it is such an number-centric specialty. So thank you very much for your attention.
Goodbye from the Hive Lab. See you soon.
Thank you so much, Dr. Keith. That was an awesome session. We'll open up the Q&A now. So please type your questions into the chat box on the right side of your screen, and I'll turn it over to my colleague, Jenny, to begin the Q&A.
Thank you, Colleen. And thanks to our speakers. We've got some questions for you. Why don't we start with Dr. Russell? I have a question for you. How long do you think it could take to create an artificial and functional pancreas with nanotechnology?
With nanotechnology, I honestly couldn't say. I think that's beyond my expertise to guess. I'm sorry.
That's fine. How about in terms of the technology we're seeing, advancing the closed loop and having glucagon as well?
In terms of how long it's going to take for glucagon, closed loop of glucagon to reach the market, the plan is to start the pivotal study for the bihormonal bionic pancreas soon, but obviously it's a long trial because it has to support, it has to provide the information for the FDA to look at the bihormonal version of the bionic pancreas, but also to support a new indication of the drug, dasiglucagon, in this intermittent, continuous use. And so they require at least a year of exposure. So that's going to be a long trial. So it's going to be a little while before that reaches the market.
Great, thank you. Turning to Dr. Peters, we have a question. Can you discuss how CGM, the advances we're seeing in CGM, can affect care for individuals in the ICU and monitoring by ICU staff?
Well, we certainly got a sense of that during COVID, but we're not yet ready for using CGM in the ICU because we have to do studies to show that if you're hypotensive or you're on pressors or on whatever else, that the values are going to be correct. So I think from COVID, we learned that you can do this in a patient who's on the ward, but the ICU portion of this, I know, needs more research that's being done, but we need to make sure that it is accurate in people in an ICU setting, or perhaps in which people in an ICU setting is it accurate. I just want to point out, by the way, that Stephen used to be my favorite geek in terms of technology, but Derek has replaced him since I saw Dave, the robot, and the drones. I think that is the coolest thing ever. But Stephen, I still think you're wonderful.
I'm going to have to somehow deal with the disappointment of falling out. It was easy come, easy go, because I didn't know I was your favorite geek. And
well, you were.
at this very moment.
You're totally my favorite geek. So perhaps you're my favorite US geek. How about that?
Okay.
You're lucky to have all of you here. It's been an amazing afternoon. Great. Turning to you, Professor O'Keefe, I was curious, in this digital health era with so many interconnected devices, how big of a concern is security for diabetes technology devices?
So yes, that's a really good question. I guess we're always going to have the threat of cybersecurity. When we think about cyber attacks around the world, we should think about it through the lens about something that's going to happen. So you heard about black swan events, things that aren't predicted and then they happen. But a cybersecurity attack is not a black swan event. It should be expected. It's part of the cost of doing business in a digital world. All the major banks, e-commerce websites, social media sites, government sites, they've all been hacked and will continue to get hacked over the years ahead. So it's about being cyber savvy, about having the right cyber defense as part of your actual architecture. And I think it's important to point out as well, it's not just traditionally the connected phone or the connected cloud. We also need to be quite savvy about medical devices being cyber secure. And that's kind of a newer concept whereby, a lot of these devices that send sugar to a pump and so on, they also can be hacked. And, it's part of the national infrastructure, as it were. Medical devices are that important. It's the same as almost like power stations. So so many people use medical devices now. They need to have the same rigor applied to them as you would for banking. And that's coming more and more into the IT industry and the med tech industry. And I said, it'll always be there. There's always going to be the threat of someone trying to do you harm. And it's just up to us to be one step ahead all the time.
Great, thank you. Dr. Peters, this question came up regarding, I believe, the devices that are non-invasive. It says, if measuring in sweat, is there any information on the lag time from capillary to sweat?
Well, on the data that's been published, there is relatively little lag time. It does better than I would have possibly imagined. So I was pretty suspicious about the whole thing. But it does seem to work, but we need to do bigger trials and we need to get more data. I also wanted to say that one of the big issues that I have is when the iOS system gets upgraded on an iPhone. I have lots of patients who are on loop, which means they're doing it themselves. And when the iOS gets upgraded, it may break loop. And so it's not just that someone's going to hack into your device. It may be the changes in your device that you may not even be aware of are going to affect its ability to function. So there's a lot out there that we're all having to learn and keep ahead of to keep our patients safe.
Great, thank you. Dr. Russell, I have a question from Miriam Tucker of Medscape. She asks, would alcohol consumption adversely affect the use of aids that include glucagon?
We don't think so, and that's based on a study that we haven't published yet, but have completed, in which we did a clamp study using glucagon, a hyperinsulinemic normal glycemic clamp study. And in addition to that, we did a ethanol clamp study. So during one clamp study, we infused IV ethanol to bring the participants quickly to a blood alcohol content of 0.1. And then in another arm of the study, we infused saline instead so that they didn't have any alcohol in their blood. And then we gave them a dose of glucagon and looked at how that changed the glucose infusion rate. And we found that there were no statistically significant differences. So the glucagon seems to work just fine, even when people are very drunk, as they were with a blood alcohol content of 0.1.
Great, thank you so much. Dr. Russell, any words of wisdom about a trihormonal pump adding pramlintide to the other two?
I think that's interesting because the pramlintide could potentially be co-formulated with the insulin. Of course, normally amylin is secreted along with insulin in a fixed ratio, and there is work that's being done to try and create a formulation of insulin and pramlintide together at a fixed ratio so that there's a basal level of pramlintide when basal levels of insulin are being infused, and then obviously more pramlintide in proportion to the amount of in the same proportion as insulin when a bolus is given. And Dr. Ahmed Haidar in Montreal has shown some nice results suggesting that that does improve or can be used to improve glycemic control. So I like that idea, and I think when there's a co-formulation that will allow what are currently insulin-only pumps to be upgraded to that co-formulation without any more mechanical complexity. And it could also potentially be used in combination with glucagon, again in a 2-chamber pump, because the insulin chamber would contain a co-formulation of insulin and pramlintide. So I think there's more work that needs to be done to show that that's worth the work of co-formulating, but the preliminary data suggests that it may well be.
Great, thank you. Are there AI-facilitated insulin delivery systems under development? How can closed-loop systems overcome the effects of the physics of obligatory oscillation?
So to answer the first question, I guess it depends on what you define as AI. The current algorithms all have some sort of adaptation or self-learning feature to them. And whether that makes them technically AI or not, I'm not sure, but I think that all of them do have some self-learning capability, and that probably will be enhanced over time. As to the obligatory oscillations, I honestly am not sure that I fully understand that question, so I might have to defer an answer on that one.
Great, thank you. Let's see, we have to wrap up. I have one more question. Feel free to weigh in, any of our speakers. What's your favorite diabetes app and why?
My favorite diabetes app is that any patient will continue to use. Everybody loves a new app, but then they stop using it. But if somebody's getting benefit from the app and it's obviously safe, then I think that's great. But whether I like it or not is relatively immaterial. It's whether a patient uses it.
I think I would say my favorite apps are probably the CGM apps. Those are the most useful. And then after that, probably apps that are helpful for doing carb counting. And there are a number of them that people use for that purpose. I think it's helpful to get instead of just eyeballing food, to have a framework to do carb counting. And so there's a number that are, I think, helpful in that regard.
My patients like Carbs and Cows, which is British, and it actually has plates. It's visual carb counting, and they can expand and shrink the plates. But anything that you can see visually, I think, helps people, and that's a tool where it being an app is useful. But I agree, carb counting is one of the hardest things people do. And then I use the dose calculator mostly in MDI patients who are using the InPen because I like that dose calculator, but people have to use it.
That's a good point. The InPen app is really a helpful one. It's, of course, linked obligatorily to that device, which is really the only smart pen that's available in the US. But it's it's really nice to be able to have the benefits of a dose calculator and the insulin on board. That adds insulin on board to the dose calculation for pen users, which I think is really helpful.
I might just add that, yeah, I agree that clinically, from my point of view, as a doctor, the best app is the CGM apps by far. When a patient comes in and then really quickly and richly, we can see all the data for the last, 7 days, 14 days, 28 days. That's an amazing app for me to have as a clinician to help people improve their blood sugars. From a patient's point of view, counting carbs, obviously, glucose monitoring, insulin monitoring, exercise, there's apps out there that can integrate all three together. The problem is that January effect is that it's brilliant for the first month, but then it kind of wanes away like gym membership. But the best app out there is probably the one that's coming, which is that if people look up Google's moonshot efforts of image to calories, where your phone can take a picture of a plate and then it can do, some AI and figure out that how many calories and carbs are on that plate. And that'll be a game changer for patients because it'll take the rote learning of carbs out of it. It won't be perfect because if you think about it, when you take a picture of a plate of food, there might be something under the sauce that the picture can't see, but it's going to make people's carb counting a whole lot more accurate, which is a good thing.
As long as carb counting is necessary, it is going to be one of the major weak points.
Yeah.
Carb counting and infusion sets, I think, are the two biggest weak points that we've got right now. And fortunately, there is more work being done on improving infusion sets as well.
Wonderful. Well, thank you to all of you for such a great discussion, and we appreciate everyone in the audience who submitted questions. But that's all the time we have for today. I'll send it back to Colleen.
Great. Thank you so much, Jenny. And if you didn't get a chance to ask your question today, or if you have any follow-up questions, you can feel free to email us at media@endocrine.org, and we can coordinate with our speakers. We want to thank our speakers, attendees, and our supporter, Lily, who made today's discussion possible. As a reminder, this event has been recorded and will be available on our 100 Years of Insulin website and YouTube channel. You can find all of our insulin resources and webinars from this past year on our 100 Years of Insulin website as well. If you're an Endocrine Society member, we invite you to log into Community Connect and continue the discussion with your peers around the future of insulin. And thank you all so much again for joining us today.